नैना को भेजो कोई। नैना come sit down. I think it's share screen. I think camera, I just sit down.
Good morning to one and all. Online participants, am I audible?
Yes, ma'am, it's audible, ma'am.
Yes, madam. Madam. I'll switch on my video also.
Distinguished guests, participants, ladies and gentlemen, Assalamu alaikum wa rahmatullahi wa barakatuh, and a very good morning to one and all gathered here. We are delighted to welcome you all to this national conference on post-COVID-19 complications, a biotechnological approach 2022, organized by the Department of Biotechnology, Jamal Mohamed College, Trichy, sponsored by the Department of Biotechnology, Ministry of Science and Technology, Government of India. Today's program will be featured by three magnanimous speakers with their edge-cutting informative lectures, followed by model presentation and poster presentation. Without any delay, let's get into the day. A person of honor and integrity never hides behind a facade. Now I kindly request Dr. S. Deborah to introduce the chief guest of the day. Good morning to one and all gathered here. This gives me immense pleasure to introduce the chief guest of today's program, Dr. Nadu Manikam, working as a senior principal scientist and coordinator of environmental toxicology, CSR, Indian Institute of Toxicology Research, Patna. Sir has completed his PhD in 2009 from Guru Nanak Dev University, Amritsar, a MTech degree in Center of Biotechnology and University, Chennai in 1990s. MSc in Botany 1998 from University of Madras with first class, and has nearly 35 years of experience in teaching and more than 32 years in the field of research. He started his career as scientist B in Environmental Biotechnology section, CSR IGRC Latina. Sir has acted as a teaching faculty from 1994 to 1997 at the Institute of Engineering and Technology, Government of Uttar Pradesh. Presently, Sir is working as senior principal scientist in environmental toxicology. He has more than 4 years of research experience at international level, particularly in Germany and USA. He has received a patent right in a process for the preparation of the hexachlorocyclohexane-free soil. He was awarded a long-term J-Star Academician Assistant CSR Fellowship Award and a German Research Foundation Short-Term Foundation Award from Indian National Science Academy. Sir has acquired various scientific skill and research experience, particularly in metagenomics, microarray, gene cloning and sequencing, restriction mapping, phylogenetic analysis, nucleic acid hybridization, pathway engineering, and microarrays. He is a life member in international and national level academic and professional society. He is a life member in German Association for General and Applied Microbiology, German life members of Society of Biological Chemists, Society of Toxicology, Biotechnological Research Society of India, and so on. He has acted as a principal investigator of various major research projects and has published more than 20 papers in most reputed international and national journals and written various manuscripts in various research projects. He has completed many research funded projects and currently is working as a national level funded research project. Such an eminent and dynamic personality is with us. I really proud to introduce him. I welcome you, sir.
Thank you, ma'am. Now I kindly request Dr. Nadesan Manikam, sir, to lecture the gathering. Please, sir.
Okay, well, very good morning. எல்லோருக்கும் காலை வணக்கம். Is my voice coming, madam? Dr. Benassi? Can you confirm?
Yes, sir.
Okay, thank you. I continue. No problem. All of you are there. Hello.
Sir, are we audible?
Yeah, you are audible. Am I audible to you also?
Sir, you're not audible, sir.
Hello. Is my voice coming? Hello. Yes, sir. Good morning once again. Are you able to hear me, please?
We are checking.
Okay, individually there should be. Okay, let's continue. Okay, can we just?
Sir, you're not audible for this technical error, sir. Few minutes.
Okay.
We are working.
From my side, it looks okay. Should I maybe log in again?
Sir, it's a fault from our side, sir. We are not connected to the speaker.
Acha, just check it and let me know, please. I'll wait.
Speaker pin irukama? Speaker pin.
Okay, I'll speak for.
To operate.
Sir, we are ready, sir. Could you please share your presentation? You can start, sir.
Yeah. Once again, very good morning. Is the voice is better now?
Yes, sir. Yes, sir.
Oh, great.
Yes, sir.
Thanks for fixing it.
Mute.
Let me check my slides. Slide is coming up or not. Huh?
Yes, sir. It's visible, sir.
Yeah. Yes, madam. Dr. Bennett. Hello, everybody. Sir, is it okay now? Hello.
Sir, yes, sir.
Slide is visible and I am audible also.
Yeah, it's visible, sir.
I am audible also. My
Yes, sir.
voice-
You are also audible.
Okay, let's go on. Okay, enough. I think there was some hiccup from both sides and we are set. Once again, good morning. Kalai Vanakkam to all students and faculties. Particularly, I'm so happy that your department is organizing such a bigger event at national level. My congratulations to the entire team, principal of the college. Yesterday, you had a great inauguration session. There were dignitaries and Dr. Benazir. I must congratulate your effort for bringing such a national important event to your college. Sure, Jamal Mohamed can be, all Jamalians can be of proud for this occasion. Okay. Is slide moving, sir? Can you see the next slide?
Yes, sir. Yes, sir.
Okay, now onwards I will not disturb you. Okay, so all the participants, you can know that we are located at Lucknow. Our institute is CSIR, under the Council of Scientific Industrial Research. It's Indian Institute of Toxicology Research, and this is where we are, and we have multiple campus. We perform. Okay. Since I saw the effort yesterday, a lot of teachers putting more, education as a continuous process. I want to wish a belated Teachers' Day, because without the teachers like Benazir and other faculties from different departments, the student would not take various professions. So I want to, because I studied somewhere near Salem, and I recall my teachers who educated in my formative years in schools and maybe colleges. Okay, coming back to our institute, IITR, 2-3 slides I want to tell you what we are doing, so that some of you may take up research as a career on environment, health, biotechnology, toxicology, biochemistry. So I want to give you a brief intro in 2 slides, our institute. So we work in the motto of safety to environment and health. Of course, our institute started working on in about 5 decades before on the occupational settings, people working at mines, pesticide industry, agriculture labor, or even other industrial setup. We were taking care of their health analysis, exposure to various chemicals like textile, dye, industrial chemical, pesticides, agrochemicals, of course, and fertilizers, etc., etc. The people who were manufacturing or even applying in the field, we were taking care of their health and epidemiological work, etc., etc. Our institute essentially works on various aspects, but essentially on environment, industrial occupational settings. I tell you that while we are working on 5 major areas, we do undertake work on national missions, like contribution to Namami Gange, which is a major Ganga rejuvenation program. Of course, a lot of startup India innovation. We have a translational hub where industries come and implement their ideas, joining hands with our scientists, working on typically, like say, food safety, environmental monitoring, safety assessment of various chemicals under regulatory regime. Because our institute has a GLP compliant facility, the students, when they go for research, they should know that good laboratory practice is a laboratory protocol which is by OECD, harmonized protocol across the globe. If you carry out a scientific experiment with a certain SOP, standard operating protocol, those will be applicable globally or in certain known countries. So we do conduct some programs, Skill India, and we train a large number of college students. Of course, there are a lot of people from Pollution Control Board, etc., etc. So we do conduct regularly. If you look at our website, in a given time, there will be one or the other courses will go on. So some of you interested may come apply and please take part in it, because these are all advanced courses, exposure to maybe highly sophisticated equipment, like state of art techniques like in vitro cell culture, 3D printing of organs, or even stem cell research, biotechnology, basic cloning experiments. So various things we impart training to students at different stages. So having told that, these are the major domain of our institute: environmental toxicology, food, drug and chemical toxicology, regulatory toxicology, as I said, GLP regime, systems and health, where we use a large number of animals: monkey, sheep, rabbit, which is now we eliminated rabbit, guinea pigs, rat, of course, vista rat, mice. So all these animals are there. We do have a GLP compliant animal facility. So we undertake a lot of effect of adverse chemicals using these systemic approaches. Of course, newly added division is where AI, ML, or even other big data management, or even various software tools to predict about the adverse effect of chemicals on humans. And of course, proteomics, genomics are part of this toxicoinformatics. Okay, so all going good so far. Introduction, students, all of you are with me. Are you hearing? Yes, anyone? Okay.
Yes, sir, you're audible, sir.
Okay, thank you. So, okay, coming to the subject theme, which is under the discussion, for which your faculties put effort. We have the funding sponsorship for COVID. Of course, specifically the theme of couple of days talk is post-COVID issues, both health, monitoring, and of course countering them and finding diagnostic tool, managing the disease better. Definitely, I don't want to go back to the panicking COVID time, particularly students and teachers. You know that second wave was so panic that maybe many of you are like me, lost the dearest and nearest ones, relatives, friends, mentors. So it was truly panicking time. You know that governments, like agency, like global agency, like WHO, like World Health Organization, international governments, joint task forces. Initially, we were not prepared at all. We understand that disease, some normal flu, fever, viral fever, all those things we have been treating effectively. Children's, elderly people, all of us were treated well by doctors. We have medicines. But initially, when this panic gripped, many governments, scientists, doctors put effort in bringing down even diagnostic kits. Large number of companies put their effort with the basic available tools, infrastructure. Like, for example, students know that two major companies like Bharat Biotech and of course the our Poonawalla company, which is Serum Institute at Pune, brought the Covishield, and Bharat Biotech brought Covaxin. So people immediately responded. Government worked proactively. We controlled. In our own state, Uttar Pradesh, in papers or media, you might have read that I know you people are aware that even relatives were not visiting hospitals where their relatives were admitted. So what happened here? It was found that people threw dead bodies, not even burning or burying them, into rivers. For example, SARS-CoV, as all of you know, this novel coronavirus WHO named it 2019 first, then they call it COVID-19, and this is zoonotic. The term is used is zoonotic, that comes from the origin of this COVID virus is animal origin, zoonotic they call. So large number of deaths, as you can see. But what I was trying, so in Uttar Pradesh, you know that major river is Ganges, which flows from upper glacier Gomukh, and then about 2,600 kilometers before it falls into Bay of Bengal. So large number of dead bodies were thrown, but we have been asked to assess the COVID's presence since large number of dead bodies thrown in the river. What is the water quality? Is the virus being detected? So we responded to the needs of national needs. What we did, like we did sampling of river Ganges, particularly in lower, Uttar Pradesh, and of course some parts of Bihar, where literally, yes, we could see some dead bodies. Of course, maybe the patients died due to COVID, or normal patients died due to other reasons. During that period, were also thrown. So we undertook detection of SARS virus. Student, maybe yesterday some of the faculties taught you like how the COVID's presence is detected through nasal or throat swab. They collect sample and test like RDPD genes or nucleocapsid N genes. There are various gene targets. Later on, once this virus was sequenced, human like genome sequence, there are various portions we can use it as a diagnostic tool for detecting yes, it is present or not. So real-time PCR basis, people detected it. So we collected a huge number of samples, as I have shown in the previous slide. Like we undertook sampling on these regions. So we collected and we found, of course, the river is so big and water flowing. We could not see actual good amount of loads of this virus. So along with that, we also detected various parameters during that coronavirus traces when we found out. We did an overall water quality assessment also there. For example, other than the COVID, we do undertake work on understanding the sewage treatments. Maybe if you students know that antimicrobial resistance, for example, again, I'll come back to post-COVID repercussions, the effects. All of you now, even students, family, medical students, everybody, even without going to doctor, during the COVID period, azithromycin, Dolos 650. There are other like medicines or antibiotics. Like over-the-counter medications, we go and we ask, and globally people use tons and tons of these antibiotics. Their effect also going to be felt in coming years. Already we have been producing, say, macrolide, beta-lactam group, lot of streptomycin groups, large number of antibiotic classes. We have been using it for one or the other purpose to control diseases. Large number of these compounds come into environment through urine, metabolites, date expired sample, unused tablets, medical waste, hospital waste. They are all thrown in a municipal dumps. Of course, some of the developed states, like even Tamil Nadu or here, Maharashtra or other states, they do better management of this severe, hospital waste, but many of them they throw. So large number of such sewage treatment plant. This is located in Lucknow, close to Lucknow. We go there for some studies. We collect samples. What we do, we assess the antimicrobial resistance. For example, large number of these resistance develop in bacteria which are not known to show resistance previously. For example, some of the microbiologists sitting here know, like say, I'm a patient, I have say urinary tract infection, some burning sensation. When we go to doctor, we give sample, they say, okay, you come tomorrow, we'll take the I will give you the test results. What they do is they take your sample, blood or urine, they plate it on some rich medium, various, of course, blood agar or other medium. They plate it and put the antibiotic disc and tell you which is the best suited medicine based on this susceptibility test. Okay, so when we went to Ganges or the sewage treatment plant or various river water system, even tap water, drinking water system, we find large number of the CC, more than around 20 antibiotics tested. This particular pneumonia, Klebsiella pneumoniae, isolated from us from Ganges river water, shows sensitivity to only one antibiotic and almost resistant to all. These are all minor susceptibility, but so such is the MDR gets developed. So coming back to the virus again, for example, various rivers in our like Talai Kaveri, if you go to Mysore, Kodagu region, even people there also take holy dip, but also in Ganges, all of you know that Maha Kumbh Mela, various holy occasions, people take a lot of dips. Many times in northern part of India, I know, I live here for more than 25 years, people get water from the upper Ganges region and store it at home. Whenever they even get some cuts and injuries, they apply also. Why? Because in the world, there are many rivers do not have bacteriophage, but Ganges is one river which has large number of viruses that feeds on bacteria as a host. For example, here, collecting a sample from Ganges river, we could isolate successfully the viruses which feeds on infectious bacteria. For example, you look at the killing effect, like all of you students know who study virology or bacteriology. There is a lytic cycle, lysogenic cycle, through which this virus enters into bacteria, multiply, blast the cells, come out, proliferate, multiply, and then again continue this process. So through which these viruses also spreads novel genes across various bacteria. There is something called horizontal gene transfer. That itself is one talk. We will take it up in some occasion. So conjugation, transfection, like transfection by this virus. So large number of these events bring in newer properties into this. But the message I am sending you here in this slide is that we isolated viruses which can kill these kind of infectious bacteria. You see, we talked about say corona or other diseases, diabetic, blah blah blah. But all of you know that Enterococcus family of virus, Enterococcus family of bacteria like Shigella, Enterobacter, Salmonella, E. coli, they kill more people every day. Children, causing dysentery, diarrhea, cholera, large number of diseases. Just not COVID killing a lot of people. Every day we talk about, for example, there are national disease directories like more than for example, any given time, like in every hour, there are some children dying due to these kind of infectious disease. So large number of phages which can counter or kill these kind of infectious bacteria, which is also multi-drug resistant, can also be isolated. Use them countering this growth of such bacteria in the maybe water system or water supplies, etc., etc. We identify and characterize this virus through TEM and SEM also. So during this water analysis, we also quantify who are all there, how much is there, what are the bacteria sitting in such ecosystem or water bodies, and how much is there, and how much is there resistant to, like say, this carbapenems, producing enzymes or even ESBL bacteria. So we identify how much are there and who are the coliforms of bacteria which is present in such water bodies. So we, for example, we also identified these multi-drug resistant bacteria. If a bacteria is resistant to some antibiotic, we call it multi-drug resistant. If say it is severely or for example, 20 different antibiotics I said are emerging newer antibiotics. They are resistant. We call them XDR or super drug resisting bacteria or super bugs. So we identify them, as you can see, large number of bacteria isolated showing a lot of resistance. What we also do is when we go to such water bodies for viral load analysis or even other water quality parameter assessment, for BOD, COD, conductivity, etc., we undertake 2-3 things. One, we analyze water quality parameters. We analyze the pathogen of what are the pathogens present in such water ecosystem, what are the viruses present there, and also we study microbial diversity, which is useful in degradation, elimination. Some of the students may be doing projects on biodegradation, metal resistance, biotransformation, bioremediation. So all those bacteria which possess degradative genes. Pathogen loads we do undertake multi-drug resistance, as I explained, metal resistant bacteria profiling, functional genes detection. Actually, when we do whole genome sequence, I want to tell you the students that any garden soil, maybe the garden which you have, if you take a gram of soil, there are billions of bacteria there. If you put it on, say, LB agar or nutrient broth or AD rich medium, you may see, say, 20 different bacteria which are visibly different. But we do undertake the extraction of total DNA, what we call metagenome, total genetic content of a given soil, a given, say, for example, you can also do the metagenome of oral cavity, saliva, urine, gut metagenome, any environment, any ecosystem you take. Your intestine contains trillions and trillions of bacteria sitting. As we are talking, the gut microbiota, understanding of them. For example, gut microbiota in our human body is inflicted in our obesity. Now, you need not to blame. You can tell your mummy, I am not, naan vandu pizza saapradanaala, burger saapradanaala gunda illa. My bacteria is responsible, you can tell. Okay, so there are gut microflora playing a role for your obesity, diseases, and of course, it has an effect on your neurobehavioral aspect also. Neurons also, there are trillions of networks with billions of neurons in our mind. Bacteria has direct control mechanism, the gut microflora we have. So any given ecosystem like soil, a rhizospheric soil of an agriculture field, oral cavity, your rectal ecosystem, gut, we can do metagenome. To understand what are the useful genes involved in metabolism. There is another term I am introducing, metabolomic also. You collect your total, bacterial population, you can collect the sample and do a metabolomic through total RNA isolation, transcriptome profiling, and metabolomic profiling also. So large number of research happening. So what we do essentially is understanding the metabolic pathways. Of course, as I said, like viruses, we detect what are the phages, plasmid, transposons play a role. So all these are important events, even if the COVID, post-COVID situation, when virus loads go into an ecosystem. That's why students and faculties, that we talk about a lot of variations and variants. As we are talking, virus replicates, changes their genome structure, like HIV virus. All of you, maybe just a couple of minutes, I will talk about HIV virus. Of course, human immunodeficiency virus. Once they detected the AIDS virus, of course, we call it commonly, the original virus mutated, and variant forms of HIV virus also known. Here also, when initially we were panicking with COVID virus, we were talking about SARS and MERS viruses. For example, SARS, severe acute respiratory syndrome virus, and MERS, Middle East, of course, virus. So large number of those viruses only mutate, form various genetic compositions, and pose a lot of challenges for medical fraternity to develop the diagnostic kit, control mechanisms. So that's how we are actually, as we evolve, virus also evolve, change their genetic makeup, pose a lot of challenges for diagnostic tools, control mechanisms, etc., etc. So it's very, very difficult situation. Like if we impact the climate and animals, we will face all these things. Like, you know, many of the students might have looked at the China meat market, or you say maybe some of the video clips you have seen. You play with all these animals, and you know there are eating all kinds of life forms. We will continue to face some challenges, but of course, as a diagnosis, as a control mechanism, other humans are involved in controlling this, helping us also. Our institute, as I said, you know, we are maximum environmental oriented. I am also environmental biotechnology, environmental toxicology coordinator. While virus is one aspect of discussion, large number of other diseases, other exposures also impart various toxicity. For example, if not COVID for last couple of years or before, we would be discussing about pollutants like plastic, microplastics, emerging pollutant like cosmetics. I think some of the students can read this, you know, like halogenated compounds, pharmaceutical, pesticides, of course, the UV filters, the creams we apply, personal care products. We think that all these are safe and beautifying. The cosmetics we use also impart toxicity, because emerging pollutants are those, for example, pesticides, metal, some arsenic, chromium, or textile dyes. All these things we think typically as a classical pollutant, but emerging pollutants are those which we did not expect they will affect us, but they do affect us. India becoming a cosmetic market, you know, everywhere you go, beauty shops, you buy a large number of course, the product. They all have the sunlight UV filter, UVA filter, UVB filter, UVC filter. There are different class of chemicals, but they do reach water bodies, environment. They in turn affect us. So we need to be very careful in discarding our domestic waste, industrial waste, mega industries. All these chemicals going to, once a chemical is produced, manufactured, they come into environment as a metabolite product after our consumption, or they are thrown as a date expired sample. So they are bound to come. So we will be under the ecological imbalance. Trophic transfer, you see, large number of these chemicals goes into water bodies or even other ecosystem. There is a trophic transfer. Algae fed by fish, small fish fed by bigger fish. So then again, human consumption, what we call it in typical toxicology term, biomagnification. A small amount of chemical built up over the food web and impart toxicity in a severe manner. Neurotoxicity, reproductive toxicity, endocrine disruption, of course, liver metabolism is affected, hepatotoxicity. So large number of these problems will emerge. So ecological risk, climate change, they are very, very important phenomena in our life, because students know, you know, if you ask your parents or your grandparents at home, the way their life was, their strength, their even walking capability, their eating capacity, their working capability, we are all very fragile. The world we have inherited, we are not passing on to you. We have created a degraded system, worst polluted ecosystem, food are contaminated, large number of artificial colors, artificial things have come. So we have damaged quite a bit. That's why globally people sat down together, large number of world leaders, including presidents and prime ministers of various nations. They brought in United Nations Development Program, Sustainable Development Goals. There are 17 SDGs. You look at the internet, go back, 17 SDGs. The first one is poverty alleviation, hunger alleviation. Second is good water for all. After poverty, food sufficiency, and good water, these are the other 7 SDGs we are working. Our institute is working on all these things. good health, of course, we work on a lot of health impacting diagnostics, toxicological research, clean water. We go to villages. Our institute develops a water filter system which is affordable, usable in a rural settings without even power. So all those water purifying systems we develop also. Affordable clean technologies, industrial growth, climate action we participate. Of course, the monitoring of water quality and exploitation of bacteria or other resources from sea, and we work on other systems like that. As I said, we do undertake not only the post-COVID monitoring system, we do, like, this is a beautiful place where even I visited a large number of times, Upper Ganges. This place is called Devaprayag, where a river called Alaknanda and Bhagirathi confluence and then gain the name Ganges. So people go even up to this higher elevation, take bath and start all these holy activities. I put it as a toxicology. The pollution we started from here itself. Upper pure, serene environment, beautiful water ecosystem. There itself we start degrading and spoiling the river. So we collect sample, work on physicochemical parameters, bacteriological load, viral load. So all these things we record, we submit it to the ministries. Ganga Water Rejuvenation Ministry, we talk to directly, Honorable Minister Nitin Gadkari ji or earlier Uma Bharati ji, now Prime Minister directly takes care of this. So we provide the measures, control measures, and actual data sets for river monitoring. I know a lot of you were from southern part. Here we need to make aware of there are 30 millions of people live all along the Ganges in this 2,500 kilometer stretch. So some of the worst pollutant states were given to us to take care of their countering mechanism and of course the monitoring system. We do undertake like pHs, polyaromatic hydrocarbon or pesticides loads, their monitoring, etc. We do undertake even during the COVID time. We did water analysis using the drones, where humanly impossible area like some of the interiors. We go deep through drone and drone based collection of samples, drone based GPS positioning, all kind of data we take, and district wise we analyze the pollutant, the pathogen, the water quality measurements, because large number of people depend on good water from these rivers regions here in northern part of India, particularly UP, Bihar, West Bengal, all these major states. We undertake, Yamuna river basin detection also and water quality assessment. Assessing, of course, the basic water quality and classify based on our analysis. Each district we analyze how do they stand, excellent, good, or poor, very poor. This is very important for maintaining the good health of the people living in this basin. It's very, very important. We do associate with the Central Pollution Control Board, State Pollution Control Board, river water quality missions and ministries. Very, very important. We do have a system, all these systems for as an alternate animal models. Of course, we use some birds, but we do have this algae, daphnia, zebrafish. I heard yesterday principal and other dignitaries talking about the zoology department there, and they want to tie up with biotechnology, zoology, biochemistry as a bigger, coordinated department for more such events to be organized in your college. I'll be happy to help in some of these, establish those facilities, have a collaborative work. There are a large number of scientists under me working in this particular area, testing for risk assessment of a large number of industrial chemicals, like preclinical studies, and assessing the chemical acute toxicity, life cycle effect, because these are all the animal have smaller life cycles. We can work on whole life cycle systems like cocoon formation using earthworm, hatchlings formation. You can measure the heartbeat of daphnia just watching under a simple microscope, the heartbeat, the eggs formation, etc., etc. So they are all OECD approved model. They do help in preliminary assessment, risk assessment of large number of chemicals. I don't want to go into detail. That is the situation we created before. I am taking little time in explaining you these pollutions, of course, virus, pathogens, and these chemical pollutants. Just if you have a couple of seconds, I will just play this for you people. If you go to even Mumbai coast or Chennai or maybe Pondicherry, if you go little deeper into the sea, this will be a situation. This is Indonesia, Bali, but most of our seas we have spoiled like this. Maybe we will take it in on another day with the environment, pollution, marine ecosystem, etc. So I'll stop it for you. Okay, so that was the situation, and we have undertaken an exercise in the marine coast of Chennai, Tiruvanmiyur, up to some mid stretch of Pondicherry. We have taken a tour analyzing the coastal plastic contamination, microplastics. Students and faculties, you know that one use single-use plastics is a big, huge menace. Even now, you know, all of you know that manjapai, we used to take it to market, is gone. People simply for the convenience, we take large number of plastics, domestic setup, industrial setup, tanks, overhead tanks at our home, and you know, from small pin pot or fan or chair, all around you. If you see, starting from your pen, pencil, chair, if you are sitting in your room, how much plastic we use. They are all create, even washing creates a lot of microplastic. The microplastic reached the human placenta, microplastic reached our table salt, because marine, you know, the purification process, since all these less than 5 micrometer microplastic is available in the sea coastal system, they're all, you know, gone into even sea salt purification system. So table salt also found to contain about 28 brands. 8 brands has microplastic. IIT Mumbai study is there. Human placenta has microplastic. There are large number of microplastic contaminations. So we did undertake along with the NIOT, we joined a team to understand, identify what are these microplastic present and what type: polypropylene, polyethylene, terephthalate, blah blah blah, are 6 major groups. What are the plastics present and how much they are present? Why I am telling, these microplastics known to take up other chemicals, viruses, bacterial particles among them and spread across the food web. You know, again, there is a risk of contaminations coming into our ecosystem. So we do undertake studies on isolating bacteria that breaks down eating a lot of bacteria. I don't want to go into detail. We have at least 7 different bacteria isolated from various ecosystems, having beautiful enzymes to eat up those plastics. We work on genomics and proteomics of it, detailed enzymes which are involved: hydrolases, hydroxylases, lipases, and other enzymes. We know the structure, we know the protein. As I am speaking to you, my student is working on protein purification, which are involved in these kind of plastic transformations. And some of the impacts, as I told, IITR develops technologies for impacting the rural folks for better drinking water facilities, etc. I think almost I come to the end. So anyway, what I spoke, you know, so I must stop here. Let us have an interactive session. I'm sorry that I took slightly deviated path today, starting from COVID coronavirus detection from the Ganges river water to some of the environmental pollutant. I want to give a glimpse of what we do, so that some of the students or faculty want to visit, have a kind of project work, dissertation, training. You can come and talk to some of my colleagues and work. As I am talking to you, some of the students from Gujarat, Rajasthan, and even other parts of the country working in my lab. So this will provide an opportunity. Any specific topic, if you show interest, I can ask my other colleagues, as I am being senior here, I can ask any other scientist to deliver a specific, for example, stem cell research, food color, food toxichemical, food safety, neurotoxicity, cancer biology, or even other ecotoxicity assessment and ecotoxicity people. All of them are my colleagues. I can ask them to deliver a talk for you. And I know Benazit madam is very active in organizing such events. I know I talked to her before also in one of the platforms. And I'm happy that large number of your teachers are very, very proactive in infusing new knowledge and making aware of current developments happening nationwide or globally. I think you should be happy, and Jamalians can learn more and more to spread yourself, acquire new knowledge. I think probably I can stop sharing the slides. Is it okay if I close the slides, madam?
Yes, sir.
I close the slides and come onto the screen. Stop share.
Yes, sir. Thank you, sir. It was really a very informative lecture, sir.
Madam, is it Benazit madam speaking or somebody else?
She's not here, sir. We are from the MC desk.
Okay. You take care. What's your good name?
My name is Afra, sir. Afra.
கொஞ்சம் louder பேசுங்க.
So can you hear me now, sir? Am I audible?
Little better.
How about now, sir?
Okay. How many of you are there, madam, in the class, auditorium or?
Nearly 200.
Here we have nearly 200 students, sir, in our hall.
They are BSc, they are also undergraduates or only postgraduate students, madam?
No, it's all BSc and MSc, and also the research scholars and other students from different colleges.
Okay. Good. So if you have interaction, keep the mic close so I should be audible. And please. Okay. Can we interact for next 10 minutes or 5 minutes? Students எல்லாம் கொஞ்சம் ஆக்டிவா இருக்கிற மாதிரி தெரியுது, ஆனா இங்க அங்க பாக்குறாங்க. யாராவது क्वेश्चन கேளுங்கம்மா.
Hello, good morning, sir.
Good morning.
Professor Dr. Sebastian from the NCDS.
Hi sir. I can see you. Yeah.
Some students are asked to ask a question to you, sir.
Pardon?
Yes, sir.
கேளுங்க, please.
Please.
Any doubt? General scientific question, technical, what are the avenues? எதுன்னு சொல்லுங்கம்மா. உங்களை இன்ட்ரோடியூஸ் பண்ணிக்கமா, பேர் சொல்லுங்க.
Good morning, sir. I am Jamal from first NAAC Biotechnology. Sir, my doubt is for how long can coronavirus last in water, in saline water?
On the lighter side, that depends on China, what they do. But lighter side. Okay. Yeah, but see, actually, fairly we could control, but முன்ன ஒரு ரெண்டு சீசன்ல பாத்தீங்கன்னா something like 20-30 years before, we used to call it something Spanish flu. It was a menace. ஒரு 2 வருஷம் ஒரு வருஷம் இருக்கும், அப்புறம் அப்படியே ஓடிடும். So we had a short-term effect of these viral loads. But this coronavirus, as you can see now, or last year we are fairly okay. Infection is there. It is not so severe. The trend we see is it may peter off, the load may go, the virulency may go. But depends on this being a new virus, எப்படி இது பிஹேவ் பண்ணுன்னு தெரியாது. இப்ப ஃபார் எக்ஸாம்பிள் ஒமைக்ரான் வந்துச்சு சடனா லாஸ்ட் நவம்பர். கொரோனா நாவல் வைரஸ்ல இருந்து ஒமைக்ரான் நம்ம we were very panicking. ஒமைக்ரான் Virologists, everybody, they panic. They immediately, the power of the biologist, biotechnology, like you are biotechnology student, we can sequence now. That is the power, Then immediately the companies which works on vaccine, they bring in such vaccines which can also. So suddenly people say BioTech, Covaxin, COVID, or even other, for example, a lot of vaccines which are in UK. Which one can counter the Omicron? So we do such a immediate stand-up, but the immediate answer to your question, how long this will prevail, it depends on only the virulence factor, mutation, and the nature. And there, the more panicking thing is how it can infect others. So there are various genetic factors, environmental factors, all these things are there. But definitely the virus behavior, it will go off. Okay, sir. Next question.
Thank you, sir. Sir, in the marine ecosystem, some microplastics are deposited in the soil. Which microorganism is suitable for degrading that kind of microplastic present in the ecosystem?
Yeah. Okay, good question, sir.
Yes, sir.
For example, typical practical based answer I give. We went to Pallikarnai, Pondicherry. There is a 1 fort. I am forgetting the name. So we isolated sample from there. Typically, we have Brucella, one Pseudomonas, and Rhodococcus. There are marine bacteria like Marinibacter. There are large number of Bacillus also thrive very well on coastal system. But the bacteria we have isolated, characterized this genome sequence where Brucella, Providencia, and Bacillus, and one Rhodococcus. They have a beautiful genes which breaks down polyphenolate and other chemicals. Fantastic.
Thank you, sir.
It
Sir,
depends on
plastic.
the system.
Thank you, sir. In the plastic polymers,
Pardon, sir?
which plastics gives a lethal effect to the human beings? Because all the human beings depends on the sea fish and seafoods, etc. Okay, through that food also the plastic contamination can enter into the human biota. Okay, which plastics is reasonable for getting lethal effects to the human being?
Sure. Good question. Because we see a lot of plastics running around, or maybe in the debris as I stood under the seawater. Yeah, actually it depends on the chemical nature. Of course, plastic polyethylene, you see, both long linear chemicals, single monomers combined as polymers, there are long chains, etc. But sir has asked me a typical question, which one? There are large number of these chemicals. For example, all of you know bisphenol. Use bisphenol in a bottle like, no, I don't have a typical water bottle. I'll just show. Water bottle ऐसा है, सादा वो वाला bottle है. So, lot of plastics, you see, this bottle is bisphenol free. Bisphenol A, bisphenol S, bisphenol F. There are all the plasticizers. Like this girl has a bottle. You see that green bottle she has in front of her, sir, behind you. Yeah, sure. Yeah. So a lot of bottle, these days we write bisphenol free. So large number of, not just plastics, the plasticizers used to harden, like bisphenols I said, were toxic earlier. But there are MHET, BHET, glycol, terephthalate. So large number of these precursor molecules known to affect endocrine, like the reproductive bodies. So sperm count will go down, egg laying capacity in birds. So large number of this. So in my opinion, sir, it depends on the precursor chemical which is used in individual plastic group. A polyurethane may be better, safer than polyterephthalate. PET bottles are more dangerous than. But that's why now leachability study people have done worldwide. PET bottles are PET leaching is more toxic than the other polyurethanes and other chemicals. Direct answer.
Thank you, sir. Sir, in the marine ecosystem, the crustacean species are the reason for trapping the many more microplastics swallowing through their food. Okay, how we can collect the microplastic from that gut region of that crustacean species? Any protocol is there? Any special method is to collect the microplastic from their gut region, sir?
Sure. Very good question. Practically, how can we measure, detect what are the debris microplastics there? There are various methods of filtering devices, plus there are dial-based methods. I will share that big protocol. I will share it to you.
Thank you, sir.
Through madam, you send me the mail ID. I will share it because it's a long protocol. There are
Yes,
quick
sir.
there are quick detection methods. There are filter methods. There are some kind of analytical tools. I will explain you.
Okay.
FTIR based analytical sophisticated tools also there. But I will share those things to you.
Yes, sir. In the comment box, I send my message, mail message, sir.
Okay. Yeah. More.
Comment box. The comment box, I send my mail, sir. Email ID.
Sebastian@gmail.
Good morning, sir. Can you hear me, sir?
Hello. Can you hear me? Anybody asking question? More question?
Yes, sir.
Can you come on to the camera side, please? Aram se, medo, cool, no problem. Yeah, I can see. Please go ahead.
Good morning, sir.
Good morning. Hey, who
I'm
is
Accredited from 3rd BSc Biotechnology, sir. My question is, how to eradicate microplastic in the marine ecosystem?
Yeah, good question. Only two possibilities. Plug the point source. We should not generate at all to begin with. Actually, government is putting a lot of effort in plugging the source. Industry should come out with biopolymers. For example, polyhydroxybutyrate, polyhydroxyalkanoates. There are large number of bacterial based or biopolymers are also being explored. So, plastic to begin with, we should stop. There are ministries playing role in banning. That's why banning plastics on this level. That's one. Second, the already reached ones. There are large number of bacteria are the one which are power industries working overnight for us to break down all those things. That's also happening. Some enzymatic process can be there, but definitely stopping the plastic use, starting from you, students like you, from your own home, we should stop it. That's one. Number two, we can also enrich by providing certain carbon sources to those marine bacteria. Like for example, oil-eating bacteria, we used to spray some fertilizers or urea to enrich those population, which will act and break down those oil-eating bacteria. Help oil-eating bacteria. So like that, we can enrich the plastic breakdown bacteria, we can enrich their population, and of course, plugging the source for reduction. So that's all the immediate solution available. But of course, we should stop using it to begin with. There are enzymatic processes also, like for example, PTAs, large number of enzymes known to be present in those bacteria. So that kind of bacteria population should be increased. For that, we should provide some certain minerals, essential nutrients, so that that population can increase to metabolize them biologically. Otherwise, large number of industries collect them, make them some kind of bricks.
Sir, you got struck, sir. Sir, it is struck. Sir, struck.
Participants, are you able to hear me?
Number side now, because she is also on struck.
You can proceed asking.
Wait பண்ணு சார் இல்ல
சார் இது பண்ணிட்டாங்க. பேர் என்ன?
Notation, sir. என்ன சார் பண்ணலாம் நம்ம சைடு
கனெக்ட் பண்ண முடியுமா?
இல்ல சார் லைக் அவரே பதிலும்
இல்ல அத யாருமே இவங்களும் ஸ்ட்ரக் இருக்காங்க நம்ம சைடு தான் ப்ராப்ளம்
இல்ல இல்ல சார் ஏன்
என்ன பண்றது?
இப்ப एक्चुअली ஆன்லைன் பார்ட்டிசிபேட்ஸ்க்கு டவுட்ஸ் இருக்கு
no no she's actually moving she's actually moving.
Excuse me ma'am.
Yes, ma'am.
Good morning. We are from Commerce College ma'am.
பாட்காஸ்ட்ல பார்த்தேன்.
One second, ma'am. Actually, sir is not in line, so we will be connecting him. One second.
Can I interact with sir? Sir.
மேம் வந்து
Okay ma'am, thank you.
டைம் முடிஞ்சிருச்சு இல்லையா? ஓகே, அடுத்த ப்ரோக்ராம் போயிடலாம்.
Okay, thank you sir. Thank you.
Thank you, ma'am. Online participants, if you have any queries, please do put it on the chat box. Thank you all.
Yeah, I have given my questions in chat box also.
Okay, ma'am. Okay, ma'am. Thank you, ma'am.
அவரே chat box very
பட் போட்டுட்டோம், இது அப்புறம் என்ன பண்றது?
Once sir gets connected, he will be replying.
Okay ma'am, no problem.
Thank you all. Now we will be having a tea break, and I request all of you to
Sir, join it.
We welcome back to the session. Online participants, is it audible?
Yes.
எல்லாருமே உள்ள
அடுத்த செஷனுக்கு சாருக்கு இன்ஃபார்ம் பண்ணிக்கலாம்.
Can you hear me?
சரி. Okay. Right.
Could you please?
The Nisha.
Could you please respond?
Right. Okay. சரி. 11:30 க்கு 11:30 க்கு நீங்க அனௌன்ஸ் பண்ணுங்க இவங்களுக்கு ஆன்லைன்ல உள்ள
Online participants, is it audible?
Yes, ma'am.
Thank you, sir. I request all of you to join back promptly at 11:30. We'll be beginning our next session with Dr. Jayakandan, sir.
Dear students and participants, all are requested to gather in our seminar hall, please.
Yes. எல்லாரும் கேம்ப் ஸ்டார்ட் பண்றது.
ஆப்பிள்.
இல்ல கிளாஸ் பண்ண சொல்லி.
Right. Chief guest introduction. Please, dear participants, please keep your class at the end of chief guest. Chief guest.
Dear participants, give your applause at the end of chief guest introduction, please. Dear participants, you are requested to give your applause after the chief guest introduction is over.
Chief guest introduction.
கொஞ்சம் வாலண்டியர் மட்டும் பண்ணிடுங்க.
Please, my dear students and participants, all are requested to give your applause after the chief guest introduction over. Please.
இது நம்ம ஸ்டூடெண்ட்ஸ் வெளிய அதர் ஸ்டூடெண்ட்ஸ் தான் வந்து
எல்லா இடத்துலயுமே இருக்கு.
பாய்ஸ் டைம் கேர்ள்ஸ்
நம்ம டிபார்ட்மெண்ட்
ஸ்டூடெண்ட்ஸ் தண்ணி குடிக்கணும்.
பயோடெக் ஸ்டாஃப் ரூம் 2 ஸ்டாஃப் ரூம் இருக்குங்க. மேல தான் ஓபன்ல இருக்கு. எப்ப வேணாலும் குடிச்சுக்கலாம். வாட்டர் கொண்டு வந்திருப்பாங்க.
There's a kind notice for you all. The other college students, we're really sorry for the inconvenience. நீங்க தண்ணி கேட்டீங்க. So வந்து bottles இருக்குன்னா நம்ம lunch சாப்பிட்டீங்கல்ல biotechnology department அங்க போய் நீங்க refill பண்ணிக்கலாம். Okay. கீழேயும் ஒரு staff room இருக்கு. அங்கேயும் we have water and also மேல. Okay. Thank you.
Our department volunteers are asked to gather the outside participants, those who are standing outside of our residential bar. Please inform them to sit in our residential bar. Please. Our department volunteers are asked to gather the outside participants standing in front of the
I welcome you all back to the session. Online participants, are we audible? Shall we start?
Just a minute.
Morning, madam.
Words are powerful. Words change lives. Words and ideas change the world. An exemplary ideal personality for the above who persuades the students' minds with his influential words. Now I kindly request Ms. Habibanisha ma'am to introduce the chief guest.
Good afternoon to one and all gathered here. It gives me immense pleasure to introduce the chief guest of today's program, Dr. Jayakanth, working as the head professor of Department of Bioinformatics in Alhappa University, Karaikudi, Tamil Nadu. Sir has completed his PhD in crystallography and biophysics from University of Madras in 2000, MPhil in physics in 1995 from MK University. Sir has nearly 12 years of teaching experience and have been in the field of research for more than 29 years. He started his career as Senior Research Fellow, CSIR, SRF, JRF in University of Madras in 1995 to 1999 and has been the researcher and the research scientist in Riken Harima Institute of Spring 8, Japan in 2003 to 2010. Presently, Sir is working as head of the Department of Bioinformatics in Alhappa University. Sir's specific area of specialization in his research includes exclusively in small and macromolecules, X-ray crystallography, and structural biology and biocomputing. His current research is on structural and function studies on viral drug targets. Under his guidance, 8 candidates have completed PhD and 8 candidates have completed MPhil, 22 candidates have completed PG projects, 8 candidates have completed UG projects, and 10 PhD candidates are pursuing under Sir guidance. And he is guiding more than 10 students with their PG projects and UG projects. Sir has published more than 175 journals in international and national journals. He presented paper in more than 80 conferences and also have acted as research person in many conferences. Sir has completed many research projects and currently he is working on BST Indo-Taiwan funded research project at the cost of 1.5 crore. Such an M person in Amit Trust. I am really proud to introduce him. I welcome you, sir.
Thank you, ma'am. Now I kindly request Dr. J. Jayakanth sir to share a few words. Please, sir.
Shall I share my slide, madam?
Yes.
Hello?
Yes, sir. Please, sir.
So it is shared, madam.
Yes, sir.
The slide, you can able to see the slide.
Yes, sir. We are able to see the slides.
Can you able to see the slide, madam?
Yes, sir.
Yes, sir.
Yes, yes. Yeah, very good morning to all. It is very great pleasure and privilege to be part of this your national conference. So I thanks to the principal of the college and also the heads of the department and the organizing secretaries of this meeting. So this is my title of the talk. Explore the novel mechanism of incubation of cell wall biosynthesis genotoxinous on effective therapeutic target. Actually, here I would like to say about that. You know the human genome project. The entire world is working on the human genome project. In that project, each country is working on a different organism. So from that different organism to identify the to solve the protein structures and propose the mechanism of the protein. Finally, it go for the drug designing aspect. So in that aspect, in India, India is working on the mycobacterial tuberculosis. So finally, it matched with the our human genome sequence.
I request you to share the screen in full view, sir. Please, sir.
Yes. Yeah, I said full screen only.
No, sir, it is not in full view.
No. Screen, full screen is there. You can able to see now. No, I didn't change any slide. I'm telling the introduction.
Can you please share the slide in slideshow, sir, please?
It is coming now.
Sir, slide is visible, sir, but it is not in slideshow form, sir.
Presentation form.
Sir, it is not in presentation form, sir.
Sir, you cannot able to.
Can you please change it into the slideshow format, sir?
Here I can see the presentation form. I said only presentation form only, not slide.
Okay, sir. Thank you, sir. You may proceed, sir.
No. Whether you can able to see the slides?
Yes, sir. We are able to see the slides, sir.
Now this is the second slide.
A text format.
Sir, it is in text format, sir. It is visible, sir. You may proceed, sir.
Visible, na?
Yes, sir. It is visible, sir.
I cannot understand, madam. Slides you see.
Sir. Hello, sir.
Yes.
Sir, your slide is not moving and it's also in a presenter view mode. Go to the slideshow mode, sir.
Then I need to come out.
Press F5. Sir, press F5 for slideshow mode, sir.
1 minute.
Yes, sir.
There is no in F5 in my.
Sir, sir, I will stop the share. I will stop the share screen. Then again, yes, sir. Share your screen, sir.
Okay.
Yeah, 1 minute. Yes, sir. Now you are share your screen, sir.
It's okay. Can you able to see?
Sir, in the tab, near animations, can you see the slideshow option, sir?
Yeah, yes.
Okay, could you please click on that?
No, whether now you can able to see the slide?
Yes, sir. We are able to see your slide, sir. We just
Right.
want it to be projected in slideshow view, sir.
Slideshow view. I don't know that slideshow view. You
It's
can.
top, sir, in the
Yes.
in the
This
top
is okay.
tab. Yes, near animations. Slideshow option is there, sir.
Slideshow option. Now we can able to see the slide, na?
Yes, sir. Yes.
Hello?
Let's start, sir.
Slide is moving, madam.
No, sir. Slide is not moving, sir.
That's a problem. You come out from that slide.
Ask him to start. No, ask him to start. Ask him to start.
Sir, close the PPT, then start again, sir.
Yes, yes, you close this.
No, actually, he's trying to. No, no. Slideshow. Western slideshow. Okay, now.
Please close.
Yeah, we have this. No, just ask him to Gmail it so we could share, right? Yeah.
No, no, please close this slide. Please close.
Ask him. Yeah, yeah. Ask. In the Gmail. On the Gmail. Oh, which Gmail? So. Convince.
Sir, share your screen now, sir.
You could send me PPT in the mail. And we would try. Please mail it and we will take it. No, sir, actually.
Excuse me, sir. Sir, for your convenience, would you make your PPT presentation to us, sir? We can take care of it.
Oh, now I will share the slide, okay?
Okay, sir. Thank you, sir.
Sorry for the
Sorry for the inconvenience, sir.
No, it's not.
ஒரு view so ஜெயகாந்தன் view பண்ணுங்க parallel ரெண்டுமே தெரியும்
Oh, can you able to see? Hello?
Yes, sir. It is visible, sir.
Visible?
Sir, you can start, sir.
It's okay, moving slide?
yes sir yes
Yes, sir.
It's okay?
Yes, sir. It is visible, sir.
Okay, thank you. This is my title of talk, explore the novel mechanism of inhibition of cell wall biosynthesis, genotoxin as an effective therapeutic target. So, as you are all well known about the escape pathogen. So, escape, it contains the 6 pathogens, including the Enterococcus faecium, and here you can see here these pathogens are notorious, responsible for the deadest hospital acquired infection, are capable of escaping the already available antibiotics. These pathogens are responsible for causing the deadest nosocomial infection, that is a hospital acquired infection. There is a question why escape is spoken widely and seriously in medical and scientific field when compared to other pathogens. Obviously, it is because of its nature to escape most of the antibacterial agent and cause high mortality rate. This escape, yes,
Excuse me, sir.
yes,
Sir, are you moving your slide, sir?
what?
Are you changing your slide, sir?
Video?
No, sir. Are you changing your slide?
No, I am not changing any slide. I will change. I am talking this slide, okay?
Okay, sir.
So you don't worry. So once you see, you can able to see changing slide now, okay na? Can you able to see the next slide?
No, sir.
Next slide.
Not moving.
It is
Not moving.
sir.
Don't understand why. But you can able to see the slide, first slide only, right?
Yes, sir. Yes, sir. First slide only, sir. It's not moving, sir. It's not go to the second slide.
Again, request the remote control of your screen like that. It is why.
At least show him the F5.
Simply you click F5.
F5.
I don't know F5.
இல்ல like sir அதான் சொல்றேன் நேத்து வந்து பிரசன்ட் பண்றது நான் இல்ல சார் அது एक्चुअली ஈஸி
சொல்லுங்க சொல்லுங்க.
So could you please send your slide? Could you share your slide at
Gmail.
Excuse me, sir. Sir, for your convenience, you could send the PPT to your Gmail ID. We will take care of it, sir.
Madam, he left from the meeting. He will join shortly.
Yeah, sir. Phone SB
எங்க போறீங்க? இங்க பாரு. எங்க போற? இங்க பாரு.
mute unmute பண்ணு unmute.
Hello? Madam,
Yes,
can
sir.
you able to whether you check whether slide is moving or not?
No, sir, slide is not moving, sir. It is on first slide, sir.
Hello, sir.
How moving?
Sir, move ஆகல சார். Please sir, உங்களுடைய நீங்க இங்க கீழ வந்து பாத்தீங்கன்னா ஒரு கப் மாதிரி பாக்ஸ் இருக்கு பாருங்க சார். 70% பக்கத்துல 79% பக்கத்துல அதை கிளிக் பண்ணீங்கன்னா ஃபுல் ஸ்கிரீன் வியூ ஆகும்.
இல்ல ஃபுல் ஸ்கிரீன்ல தான் இருக்கேன் நானு. ஃபுல்
இல்ல
ஸ்கிரீன்ல
இல்ல அது வந்து ஸ்லைட் ஷோல வரல.
Excuse me, sir.
டெக்ஸ்ட் பண்ற மாதிரி
சார் ஒன் மினிட் சார். சார் ஸ்கிரீன் மட்டும் ஸ்டாப் பண்ணுங்க சார். ஸ்டெப் பை ஸ்டெப்பா பண்ணிடலாம் ப்ரோ சார்.
ஓகே.
எஸ் சார். ஸ்கிரீன் ஸ்டாப் பண்ணிடலாம் சார் ஃபர்ஸ்ட். ஷேர் ஸ்கிரீன் கொடுத்ததுக்கு அப்புறம் ஸ்கிரீன் ஒரு ஆப்ஷன் இருக்கும் சார். அதை செலக்ட் பண்ணுங்க சார். உங்களோட டெஸ்க்டாப் வியூ ஆகும். சோ பிபிடியை க்ளோஸ் பண்ணிருங்க சார்.
உனக்கு தெரியாது என்ன இங்க பாரு.
எஸ் சார். இப்போ ஷேர் ஸ்கிரீனை கிளிக் பண்ணிட்டு
ஆமா
ஸ்கிரீன் ஒரு ஆப்ஷன் இருக்கும் சார்.
ஷேர் ஸ்கிரீன்ல வந்து
ஷேர் ஸ்கிரீன் கிளிக் பண்ண உடனே மேல ஸ்கிரீன் ஃபர்ஸ்ட் ஒன்னு இருக்கும் சார். ஸ்கிரீன்
ஆமா ஆமா ஆமா
அதை கிளிக் பண்ணிட்டு ரைட் சைடு ஷேர்னு இருக்கும் சார். அதை கிளிக் பண்ணிருங்க சார். எஸ் சார்.
ஓகே.
இப்போ எங்களுக்கு இதை மினிமைஸ் பண்ணுங்க சார். எல்லாத்தையும் மினிமைஸ் பண்ணுங்க. எஸ் சார். இப்போ பிபிடி ஓபன் பண்ணுங்க சார்.
மாடலா
இப்போ மேல ஸ்லைட்ஸ் போங்க சார். மேல மெனுல அனிமேஷன்ஸ் பக்கத்துல ஸ்லைட்ஸ் இருக்குல்ல சார்.
ஆமா
கிளிக் பண்ணுங்க சார். Yes, sir. Okay.
Yes, sir.
Thank you, sir.
Yes, sir. Thank you, sir. Thank you.
All that.
Now it's moving, sir. Go ahead, sir.
Okay.
Thank you, sir.
So here you can see this what is this escape pathogen? It encompasses 6 pathogens, including enterococcus like Acinetobacter baumannii, Pseudomonas, like that. These pathogens are responsible for causing the disc nosocomial infection, that is like a hospital acquired infection. So here there is a question by why we are telling about the escape is always widely and seriously in medical as well as the scientific field when compared to the other pathogens. Obviously, it is because of its nature to escape most of the antibacterial agent and cause high mortality rate. So this escape includes both the gram-positive as well as the gram-negative pathogens. In the current 21st century, the multi-drug resistant bacteria are one of the most important current threats to public health, and the escape group includes most of the serious, the MDR, the multi-drug resistance organisms. So the major drug resistance pathogens among the gram-negative members, the escape is Acinetobacter baumannii. Also, Infection Disease Society of America has declared it is as a great threat to human health. Hence, we have chosen this pathogen for our studies. Here you can see here. So this is like a rod-shaped and non-motile aerobic bacterium. It causes significant nosocomial infection, and it has highest mortality rate, like more than 52%. And here the Center of Disease Control has stated it is as a red alert pathogen, because here we can see here it's a n number of diseases happening in the picture you can see here. It causes upper and lower respiratory infection and wound and soft tissues infection and the urinary tract and bloodstream infection like this. So here you can see this contains the mechanisms like antibiotic resistance mechanism. Here you can see here how, so because of multi-drug resistance, you can see here once you are taking any kind of drug like antibiotic, you can see here the active efflux. So suppose once you enter into you enter into this antibiotic, then it is automatically sent out because of this multi-drug resistance. And also there is one thing like target site modification. You can see here we are targeting on track, but it is not binding the exact place. So like that is it is also doing in the inside the cell. And also some other things also happening the enzyme modification. Because suppose you are enzyme modification, what it will do now that antibiotic you are taken, it will degrade everything, it will break everything. And also this will do also like it will change the another kind of enzyme like this. So this is the mechanism of like antibiotic resistance mechanism. That's why. So here you can see here the Acinetobacter baumannii was various mechanism to prevent the cell from the action of antibiotics, such as active drug efflux like this. That's why it is most of the existing antibiotics has become the ineffective against the Acinetobacter baumannii strain. Like this, whatever new antibiotic scientists find, the evolving resistance is giving a challenges to the scientific community and limiting their usage. So we thought of finding some alternative to the antibiotics. Here you can see here, whatever we give from the outside the cell is developing the resistance. Why can't we find something naturally present in the pathogen itself to induce the cell death was our question. So based on that, then we came to find about the taxine anti-taxine system. Here you can see here there are 8 different types of taxine anti-taxine system, the type 1 to type 8 bacteria. The Acinetobacter baumannii has the 15 taxine anti-taxine proteins. These taxines play a role by inhibiting the translation by RNA degradation and terminating cell wall synthesis, etc. One among those taxine is a genotoxin. There is not much study about the role of it, Acinetobacter baumannii. So based on that, so we have come to the here you can see here the genotoxin. Here you can see here how it is like the promoter is binding into the plasmid. So if like epsilon and the genotoxins, once it is bind, it is process the enzymes like epsilon antitoxin and genotoxin. This will bind into together, and once it is both are in together, it is an inactive stage. Here you can see here the expression of taxin and the antitoxin in the bacterial system. So based on that, so here, like you can see here, this mechanism that the bacterial cell stable maintains the like copy number of plasmids through the PSK mechanism. So when a plasmid free daughter cell arises upon cell division, this mechanism gets activated. This in turn triggers the antitoxin quickly with the help of the protease and by the addition of a ATP molecule. The degradation of the antitoxin free, then the genotoxin and makes it a active stage. So here you can see the mechanism how once the protease cleave that one, then it is a epsilon antitoxin degradation and the genotoxins become active. So based on this, the genotoxin goes and target cell wall biosynthesis. So based on this mechanism, the next here you can see here the peptide glycogen biosynthesis pathway. Here initially you can see the for all the bacterial cell wall gives the strength and protects the cell wall from external environment. Genotoxin targets the initial precursor, uridine diphosphate acetyl glycomycin, that is called the UNAG for cell wall biosynthesis. Once the genotoxin activates the UNA kinase protein, the UNA kinase transfer a phosphate in the ATP by a phosphorylation mechanism to the OH group in the C3 position of UNAG. The result in the formation of your death end product of the UNAG3 third position. Once the UNA3 third position cannot be utilized for cell wall biosynthesis, hence the cell wall biosynthesis stops, and this would help in the controlling the pathogen. So based on this, so we want to study about the computational aspect. So we want to study this mechanism computationally and also structurally. Hence we applied the molecular docking and the dynamic study to this pathway. So here we have taken the sequence of the genotoxin. It contains the 360 amino acids. So then we take this one for molecular modeling using because of less similarity. So then we have taken this sequence. We went for the Robota server to model this protein. Here you can see the C terminal, N terminal. There is a maximum. The structure is to have the helix structures and only some beta strand. You can see here. And based on that, once we had modeled this structure, we needed to go for the validation of this model by using the Ramachandran plot. So here you can see all the residues and everything is the allowed region. So once we had done this structure model, then we have taken for the molecular dynamic simulation. Here you can see that simulation of the model. We use the 100 nanosecond genotoxin protein. Then you can see the stabilization of the model. The blue color is showing the dynamics RMSD profile of the genotoxin, and you can see the red color. It is RMS fluctuation. There is some residues are fluctuating in the active site pocket as well as the C and the N terminal region. And after that, so we needed to identify where the genotoxin is present in the plasmids. As I said before, then we used the dblast, use the protein sequence of genotoxin, and it shows the 100% identity with the plasmid. So here you can see the various plasmids are present in the genotoxin, in these plasmids. Then we compared each one, how much of the percentage the similarities present in the genotoxin. So we identify, use the D-plast. So here you can see the percentage of the genotoxin present in the plasmids. The same way, we also checked this antitoxin using the Tasmania finder. There is one kind of tool. So we have taken that sequences, then we checked that where it is antitoxin find. Here also it is showing that there is a 4 plasmids. These 4 plasmids also have the antitoxin. So based on this analysis, we have taken for the already reported structures for our studies. So here you can see here the similarities between the genotoxin of Acinetobacter with the Neisseria gonorrhea structure. It shows the 17% similarity and the score. This already deposited structure is available in the protein data bank. It contains like 8 chains. Here you can see here the chain A shows the antitaxin, and the chain B shows the taxin. So here this both the protein is taxin and the antitaxin is binding to here for easily you can see here. So based on that, so we must confirm with our mechanism. So here you can see here, then we have taken our protein, genotoxin, and superimposed with these chains. Here you can see the RMS deviation of 5 angstrom superimposed. That is only the core region of the genotoxin of Acinetobacter baumannii with the reported structures. Here you can see the red color is one and the green color is epsilon toxin. So based on that, here also we needed to identify, because we have modeled the structure, so we need to identify the active site residues of this protein. Then we have used these many servers, this online servers. There is 4 servers we have used, and each server is listed the active site residues. Here you can see the active site residues using the residue depth and the Cas-P and the 3 ligand site and everything. And based on this, we come to the conclusion that which are the residues are similar residues are present in all the tools, the server, active site prediction server. Then we selected these active site residues for the further analysis. Based on this active site residues, then we have taken the genotoxin protein and also the antitoxin, epsilon antitoxin. Here we went for there is a HADOC server, protein docking of the genotoxin and the epsilon antitoxin using the HADOC server. Based on that, here you can see here the van der Waals energy, electrostatic and dissolution energy. So we come to the conclusion we have taken based on the score as well as the interaction between the genotoxin and the antitoxin. Both the interaction residues are showing here. So, and based on this one, so then we have done here molecular dynamics of the genotoxin and the antitoxin. Here you can see here after the 80 nanosecond. So here you can see this both are binding into the each one well binding. After 90 nanosecond, here you can see the genotoxin as well as the antitoxin is separated. You can see the separated from the after 90 nanosecond. Genotoxin is separated and antitoxin also separated. So based on this mechanism, then also we want to confirm these things with our experimental result with the available structures. We have taken the 2 organisms. One is the gram positive, another one is the gram negative. Also here you can see the RMS deviation between the genotoxin and the antitoxin. There is after the 70-60 nanoseconds, it is both are separating. So then we confirm that. So the mechanism, it is similar with also whatever we predicted is compared with the reported one. So then based on this, we have taken to find this is it contains the ATP binding site. ATP binding site that we needed to identify that particular motif of this protein, then we have used this conserved domain architecture retrieved tool, and also we compare with the our sequence, data structure sequence with the reported structures. We identify that is a region. There is a P-loop region that is called the ATP binding, the nucleotide binding motif. So based on this, we come to the conclusion this particular places this ATP molecule will bind like that. So then, based on this analysis, we have taken our both the structures like genotoxin and also antitaxin. Here you can see here the ACK motif which we find from that tool. So then we have taken the ATP molecule, then the ATP molecule docked into the taxin and antitaxin protein. Then here you can see where it is binding the ATP molecule based on the molecular docking of methods. So after that, here you can see these are the like interactions of ATP with the taxin and the antitaxin. Here you can see the interaction residues. So there is a like serine 56, lysine 58, and glutamine 106, and glutamine 347. These are the residues. The active site residues are interacting with the ATP molecule in the active site packet. So then here you can see, so again we want to confirm this mechanism. Like now you can see here, this is a taxin is there, antitaxin is there, as well as the ATP molecule is there. So we checked the molecular dynamics of this all together. Here you can see here the fluctuation between you can see here the 10 nanosecond, there is no any kind of moving out. So you can see here 10 nanosecond, the ATP molecule is there and antitaxin is there. And also you can see here the 20 nanosecond, the ATP molecule and the genotoxin is there, but antitaxin is away from the 20 nanosecond. If you go to the, again, you go to the 30 nanosecond, it is coming together again. It is temporarily, it is going to binding the genotoxin, antitaxin and the ATP molecule. You can see here 10 nanosecond how it is absorbing, and 20 nanosecond is how it is happening, and 30 nanosecond is how it is happening. Then again you come to the like 40 nanosecond, it is coming out the antitaxin. So it is like this kind of mechanism, like antitaxin was expelled from the genotoxin, influenced by the ATP, which was observed at the 47 nanosecond. Later, the ATP was automatically eliminated from the genotoxin and enters to the UNA kinase. As we have seen the mechanism, then it is coming, it is entered to the substrate binding packet. Here you can see here, after the 15 nanosecond, you can see here the genotoxin is separately is there, and antitaxin separately is there, and the ATP molecule also is there. And further, what it is doing, the ATP molecule again, it is going to make into the another involved into the another kinase protein. Here you can see here. Then we want to do the molecular dynamics of all the together, like genotoxin, antitoxin, ATP molecule with the gram positive and gram negative. Here you can see the same mechanism is happening. So we want to cross check everything whether it is compared with our reported with our present studies. So based on this, so here what we have taken, as I told, that the ATP molecule is involving to the UNAG kinase. So here you can see here, then we have taken the UNAG kinase. Then we constructed, this contains the 517 amino acid. Then we constructed the model. Here you can see also the structure of this model. Then again we have checked with the Ramachandra plot, the allowed region, everything. So based on this, again we have done the molecular dynamic simulation of the UNA kinase. So here you can see this molecule is stable in the 15 nanosecond, as well as the fluctuation of the active site packet. The residues are slightly fluctuation. And then, so again we want to confirm everything with the reported one. So then we have done the multiple sequence alignment of our Acinetobacter and other reported genotoxin and UNA kinase. Here you can see we also here observed that P loop region, like SGS loop, like ATP binding place, SGK packet. So based on that, we have taken this structure, then we have further analysis, like we have taken. For here, you can see here how it is like molecular docking of ATP to UNA kinase, like genotoxin, the ATP molecule and antitoxin is temporarily favoring us. As I mentioned before, it is a 30 nanosecond, it is coming outside, like temporarily binding and coming out in the 40 nanosecond, it is everything separated. This ATP molecule further is involving into the UNA kinase. So here you can see here the UNA kinase also we modeled. Then we docked the ATP molecule. Then we have taken our genotoxin from the acinetobacter protein. Then we have superimposed. So here you can see here with the UNA kinase ATP to the genotoxin. So exactly it is the entering into the active site pocket of the UNAG molecule. So based on this, here you can see the how it is the molecular docking of the UNAG to the UNA kinase ATP. That is a UNAG kinase ATP molecule. That is a UNAG kinase. Inside the active site pocket, the ATP is there. There is a UNAG. That is a UNAG is a substrate. That UNAG again it is binding into the, as I told you before, the ATP molecules taken in one phosphate group. It is forming in the third position of the that UNAG. So here you can see the how it is binding to the ATP UNA kinase and the ATP UNAG. So based on this analysis, here we find that the docking score as well as the like the energy of this binding energy. So we have confirmed this everything is binding properly, and based on this, we come to the we need to confirm our mechanism. Here you can see the before before in the beginning itself I mentioned that there is a UNA is a substrate, and you can see that it is taking the ATP, and once it taken that that phosphate group is in the forming in the third position of the UNAG. That's why the UNAG the third position. The same way another thing also it is happening. So here it is binding into the fourth position UNAG. So we have needed to confirm whether it is which one is very strong. The based on that we have taken this one and we docked with the UNA kinase enzyme UNA kinase. The UNAG 3P and as well as the UNAG fourth position we docked with this one, and based on the score we have taken the UNAG kinase third position molecule. You can see here minus 4.8 kilo calorie per molecule. That is for fourth position. It is showing that minus 2.4 kilomole. So based on that, we have taken this one. And next, what we have done, then we need to do the dynamics of this both things. We have done the dynamics. Here you can see here, and the red curve is showing the UNAG third position, and block is showing that the UNAG kinase is everything is stable. This is the diagram, the figure B is showing that the RMS fluctuation of the both the UNAG kinase as well as the UNAG third position of the substrate. So here is you can see here the mechanism how it is happening. This mechanism you can see here genotoxin protein and antitoxin protein. That is genotoxin is showing that blue color, and antitoxin is showing as the green color. Then as I mentioned before, based on the dynamics, it is coming out it is separating the like genotoxin and antitoxin. Here you can say then once the ATP molecule bind, then it is together it is forming like a genotoxin, antitoxin with along with the ATP molecule. So once the ATP molecules, as I said, the ATP molecule is temporarily is coming out. It's after the 47 nanosecond. Then then that one is entered into the UNA kinase. So here we have modeled the UNA kinase protein. Then again that ATP is binding to the UNA kinase. Then again we have bind to the that substrate UNAG. So you can see here the all together, and it is binding in the pocket. You can see the interaction of the UNA kinase ATP and UNAG, which is binding to the third position of the that UNAG molecule. So this is the mechanism how it is happening. So this we have done everything in the computational aspect. So based on this computational aspect, so we have taken this mechanism to go for the very wet lab oriented studies. So then we plan to do the experimental aspect. This is the final mechanism, like we can see here, as I mentioned here. There is a like the highlights of the study is once understanding the plausible mechanism to identify the free energy of ATP UNH complexes, and the second one is to identify the specific interaction for the corresponding genotoxin. And the overall from the study, it is clear that genotoxin is temporarily favors the ATP molecule, temporarily favoring the ATP, and it stimulates the NAAC kinase to end the cell wall biosynthesis. So the result illustrates the role of genotoxin, which will induce the bacteriostatic activity to prevent the peptidoglycan biosynthesis. So therefore, the predicted result catalytic roles of genotoxin, which will induce the bacteriostatic activity to prevent the peptidoglycan. So this is the mechanism we want to confirm into our wet lab oriented. So that's why we are planning to cloning and expression and purification of this protein, and make a single crystal and collect the data of that single crystal data, and then solve the structure, and again we can make the complex of this proteins, both the pyridoxine and antitoxin as well as the ATP molecule. So after that, so we want to solve the structure from the using the X-ray crystallography. Here you can see now this work is under progress, the cloning of that enzymes, and then expression, and from there we will purify the protein, and once purify the protein, then we will make the crystal, a single crystal, then use the X-ray diffraction, and using this X-ray diffraction, then solve the protein structure of this genotoxin and antitoxin, and make the complex of this one, and then propose the mechanism as we predicted in the computational aspect. So based on this, our study is going on. And here, as I want to tell about, here we are giving the Master of Science in Bioinformatics as well as the Master of Philosophy in Bioinformatics, and we are giving the Doctor of Philosophy in PhD. So those who are interested, maybe you can approach our department, and you can join us here. I am master students or doctoral PhD students. These are the facilities available in our department. These are the facilities. So you can see the computational facilities as well as the wet lab facilities. So you can do whatever we are predicting the computational aspect of any disease related protein, that will be done in the experimental aspect and through the mechanism. And finally, we take that one for the drug designing aspect. So this is our facilities. And also, I would like to acknowledge these are the funding agencies supporting for our research as well as the infrastructure development aspect. And these are all my students. Each students are working in different proteins, and also working under postdoctoral fellow. This genotoxin protein and antitoxin protein, this Karthia is working her PhD program. So she is the only have done this work. And this is our science block. This is our science block. So this Department of Bioinformatics located the 4th floor. So whenever you find, or you also, we are also encouraging the students for do some projects, MSc projects, like summer project, as well as the like 6 months project also. If you are interested, maybe you can also join with, come to the visit our department. So thank you very much.
Thank you, sir. Thank you. It was really very informative lecture, sir.
So I'm sorry because it was too delayed because of this connectivity.
It's okay, sir.
So maybe
Now it's time for questions. Is there any questions from the participants? Online participants as well as offline participants.
Excuse me, sir.
yes, please.
Good afternoon, sir.
Yes,
My question is, where can we isolate zeta substance? Isolate large amount from the living organism.
I didn't get it properly.
முன்னாடி வாமா பாப்பா come friend
I didn't get it properly,
one
please.
second sir இங்க வந்து பேசுங்க கேக்குமா இப்போ yes
Sir, where can we isolate zeta substance? Isolate large amount from the living organism.
See, large isolating, you mean you are asking isolating large amount of genotoxin or
yes sir
Yes, sir.
because for large amount for we want for making the protein crystal. That's why we need the large volume of isolation and get the protein more volume. Based on that, we need to go for the screening. So until to get the crystal. So that is your question, or?
Yes, sir.
So based on that, because we have the n number of screenings are there, more than 4,000 screening conditions are there. So we need to screen. So we need a large volume of protein until get the protein crystal. That's why we need a large volume.
Thank you, sir.
Good afternoon, sir. My question is, you said at particular nanosecond, the toxin and antitoxin complex is detached. What just happened? What is the reason behind this? Will it happen in the living system too?
That's why it is happened in the living system. That's why we want to do it in the cell wall itself, the inside itself, because genotoxin, antitoxin is once it is binded, it is cell wall biosynthesis it will start. That's why we have taken the initial protein like UNA kinase, and it will block, it will stop the cell wall biosynthesis, that is peptidoglycan glycosid biosynthesis. So that's why we have taken that one. We need to stop that one. So we have taken for that. So we have done the everything in the computational aspect. It is a predicted method. It is a computation. It's not a 100% accurate method. That's why we have taken the experimentally once proved, then we can say everything very properly.
Thank you, sir.
Good afternoon, sir.
Yes.
Myself, I am Neelani from Tiruchirappalli College, sir. My question is, which enzyme involved in zeta-toxin?
Yeah, like that is a UNA kinase are involved. That is UNA kinase. That enzyme is involved in UNA kinase.
So one more question. What are the factors that affect the zeta potentials?
What are the
Factors that affect the zeta potentials?
factors? I don't understand. What are the factors affecting the genotoxin? So this is like this is like as I told, this is a pathogen related. So like you are a hospital, long staying in the hospital, it affect the pathogens. It's like infection from the bacteria. So there is already the available antibiotic drugs are not efficient. Not efficient. It is a multi-drug resistance because as I told you, so whatever the available antibiotics, everything is now multi-drug resistance. So whatever you take, it won't kill the bacteria. So that's why we need to go for the new antibiotic. New antibiotic. In the year, the most of the 6 escape pathogen, there is a Acinetobacter baumannii only more than 52% was the mortality rate is high. So that's why we need to identify the new antibiotic. Here we are doing the computational aspectly. We are making the how it is working the mechanism. Once that one is mechanism is stable, then we will go for the designing of the new drug. So
Okay, sir. Thank you, sir.
Thank you, sir.
Next question.
Yes.
Thank you. Very good.
Good afternoon, sir.
yes.
My question is, how can we use cetotoxin to the human beings?
The data given already it is there in our human. So all the things are there in the peptide glycation biosynthesis. Like so that's why we want to try our cells, our system itself. So actually, whatever we are taking the antibiotic, it is not accepting because of multi-drug resistance. That's why we want to do it in the naturally. We need to stop the peptide glycation biosynthesis. That's why we have taken this one. So that is the our study.
Thank you, sir.
Is there any questions from the online participants?
And there is no question. Anyway, thank you very much.
Sir, there's a question from one of our participants, sir. They've asked to give the suggestion on best docking online tools, sir.
This AutoDoc is there. AutoDoc is there. So freely available. You can download. Hello.
Okay, sir. Thank you, sir.
One more thing, madam. So we are conducting the conference in the November 21st to 25th. There we are giving the 2 days workshop on computer aided drug design, docking and dynamics and everything. So that is giving by the Sodinger. If your students are interested, maybe you can join that conference so they can learn the hands-on training. The hands-on training.
Sure, sir. We have already received the invitation, sir.
Yeah, so maybe I send. I think so. So
Yes, sir. Thank you, sir.
maybe you can send your students also.
Yes, sir. Sure, sir. Sure, sir.
I am very sorry for delay and due to the connectivity, everything. Maybe if it is come directly, maybe it may be very good interaction.
Yes, sir.
Next time, if
Thank you for your time, sir. It's okay, sir.
if any chance comes, I will come over there and I will do the.
Sure, sir. You're welcome, sir. Thank you, sir.
Thank you very much, madam.
Thank you.
Shall I disconnect, madam?
Yes, sir. Yes, sir. Thank you for your time, sir. For the online participants, now it's time for the lunch break.
We will inform later.
Regarding the next lecture, we will be getting your notifications. Thank you. Thank you for your time.
Hello, good afternoon, ma'am. we get please be ready for the presentation. Participants, please be ready. Raffles. Online, but
Good afternoon, online.
Online participants. So 4 minutes of
Good afternoon, all. Welcome you back.
4 minutes on the.
We'll be starting our online poster presentation in 4 minutes. Please get ready, participants. The time duration for your presentation is 4 minutes for your presentation and 1 minute for your discussion. So it should be 5 minutes in total. Please stick up with your time.
Bell Mario.
Am I audible? Online participants, could you please reply? Are we audible?
Yes, ma'am.
Okay, thank you. I repeat, the time duration for your poster presentation will be 3 minutes for the presentation and 1 minute for discussion.
Shall we start? Start. Nago. First participant. Listing.
So here we begin the online poster presentation. Now I call Isa Sharma, participant number 1.
Yes, ma'am.
Are you in line?
Yes, ma'am. Ma'am, can I share my screen?
Yes, please, sir.
Is it visible?
Yes, sir. You may start, please.
Yes, the title of my post, this is a poster of the proposed work. The title of my poster is Energy Homeostasis Related Genetic Markers and Pathogenesis of Metabolically Healthy and Metabolically Unhealthy Obesity. So first of all, obesity has become a global threat to mankind. It is contributing to diseases like coronary heart diseases, hypertension, type 2 diabetes mellitus, and hyperlipidemia. Obesity is a condition that is controlled by many factors, and genetics and energy homeostasis plays a very important role in the disease outcome. Still, the association of different genes like FTO, MC4R, leptin gene, adipose gene, etc. with obesity and with other metabolic abnormalities is still not clear. So there are some important points which we need to give our focus on. And first is defining and classifying obesity, because we only think that obesity is all about fat accumulation, but no, obesity is also important. Fat distribution is also important in obesity, because there are certain organs in which fat is accumulated more, like in liver or pancreas. It will lead to the more metabolically unhealthy obesity. And gut-brain axis and energy homeostasis hormones, this endocrine system is also inefficient in obesity, and we also need to focus on the gut-brain axis. And then metabolic tissues undergo energy misbalance and the modified paradigm of the signaling molecules in obesity. And beyond the energy balance, imbalance, psychological effects are also a factor of obesity, because whatever we are having in our diet or whatever activity we are doing our whole day, it affects our psychological status. And if a person is having sleep shortage or any psychological mood disorders like depression or stress, it will lead to obesity. And it is also vice versa, because obesity also leads such conditions. In this diagram, you can see that our gut is associated with our energy balance, and it also affects the neuroendocrine signals. And the brain also affects the neuroendocrine.
Can you please come to the conclusion, sir?
Yes, ma'am.
Can you please come to the conclusion?
Yes, ma'am. Yes,
ma'am. Sir.
So The conclusion of this study is very important to determine the association of gene polymorphism with obesity in the study population. Also, when combined in haplotypes, a synergistic effect will be observed in leptin levels and obesity risk. Major controllers of energy homeostasis in obesity are leptin and leptin receptors. And there are these 5 hormones, which are gut-brain axis and energy homeostasis hormones. And their signaling also affects the energy metabolism. These are ghrelin, glucagon-like peptide 1, peptide YY336, cholecystokinin, and amylin. So the level of these hormones should also be checked with the single nucleotide polymorphism of the same genes. And this is the methodology which will be followed for this study.
Thank you for presenting the poster, sir. How many persons you have used as a sample?
Sir, you are not audible.
How many persons you have collected samples from?
Sir, yes, sir. I have collected a sample of 600 patients and 1,000 controls. In 600 patients, we have different categories of metabolically healthy obesity, metabolically unhealthy obesity. The metabolically healthy obesity patients are those patients which are visibly obese, which have higher BMI, but they have normal lipid profile, they have normal cholesterol status and normal blood pressure status. But metabolically unhealthy patients are those patients which are visibly obese, but they have high lipid profile, high cholesterol level. They are also have diabetes mellitus and from high blood pressure also.
How do you have control group?
Sorry, sir, please.
Control group. People without.
Sir, in control group, we have taken the population of individuals which are not visibly obese, which have normal BMI, and they are also not having any disturbed lipid profile, any blood pressure symptoms, or any symptoms of the diabetes.
Does your hormonal levels obey any expert test, or have you done any comparison test?
So you are not
comparison test like T test, chi-square test. Have you done any statistical analysis?
Yes, sir. This study is still going on. Actually, I have recently joined my PhD, so this is a kind of proposed work. We have not completed the work. It is still going on a sample collection.
Okay, thank you. We move on to the next step.
Attention, please. The online participants are kindly requested to listen to attention.
Okay, ma'am.
The time duration for poster presentation is 3 minutes, and the first reminder alarm will be given at 3 minutes. And then the 1 minute is for discussion, and the 4th alarm will be given at 4th minute. You have to conclude with it. Next I call Priya Dixit. Are you in line? Miss Priya Dixit, slot number 2. You may start. Miss Priya, can you start?
Yes, ma'am. Yes, ma'am.
Your poster is not visible.
Hello.
Yes, ma'am.
Ma'am, please share my PPT presentation.
You're not able to share?
Yes, ma'am. Yes, ma'am. On WhatsApp group.
வாட்ஸ்அப் இருக்கா இல்ல இல்ல எனக்கு அனுப்பி
Okay, ma'am. You just send us and we will now to the next participant. You may do it later. Okay, Ms. Priya?
Ma'am, I am already sent it on your WhatsApp group. Please check and.
Yes, ma'am. Yes, ma'am. We need some time to work on it. So next I call Ghazala Shaheen. Are you in line? Slot number 3.
Yes, ma'am.
Yes, you may present now and start.
8283.
மேம் அவங்களே விசிபிள் ஆயிடுச்சு விசிபிள் ஆயிடுச்சு சரி சொல்லுங்க
Ms. Priya, are you in line?
Hello. Yes, ma'am.
Yeah, we can see your poster now. You may start.
Okay. Okay. Thank
Yes,
you, ma'am.
yes, you may start. Yeah.
Good afternoon to all of you. Myself, Priya Dikshit, Research Scholar, Department of Biotechnology, Era University, Lucknow. Today, my topic is lack of association between the osteocalcin in third gene polymorphism with BMD in North Indian postmenopausal osteoporotic women. First of all, I would like to tell what is osteoporosis. Osteoporosis is a multifactorial skeletal disease characterized by bone mineral density and increased fracture risk, and it is growing health problem. According to International Osteoporosis Foundation, it has been estimated globally more than 200 million people are suffering from osteoporosis. It is more common is female as compared male with ratio 2:1, and particularly in postmenopausal women. The human osteocalcin gene is located in 1q2531 chromosomal region, and genetic evidence suggested that osteocalcin is a candidate gene. Osteocalcin is a member of non-collagenous protein family, also known as a bone gamma carboxyglutamic acid is found in bone. It is exclusively produced by osteoblasts under transcriptional regulation through a vitamin D response element, and is well known as a marker for differentiation, mature osteoblasts, and a determinant of bone calcification process. Osteocalcin may also be involved in the recruitment and differentiation of osteoclast precursor at the bone resorption and bone remodeling. Doi et al. Identified a polymorphism in osteocalcin gene with the Hin-3 restriction enzyme, and so it is also known as the osteocalcin Hin-3 gene polymorphism. The material method of this study, firstly, we take the ethical clearance from Institutional Ethics Committee, then the subject inclusion and exclusion criteria on the basis of WHO, obtained written informed consent from all subjects, collected 5 ml blood sample from patient and control group. The BMD was measured by dual energy X-ray absorptiometry. Then after DNA extraction from red characterization of the DNA, all subjects were genotyped by PCR-RFLP method, and the PCR amplicon were analyzed by separating 2% agarose gel and visualizing by tritium bromide staining. Then statistical analysis was done by SPSS version 16.0. All p-value below 0.05 were taken as a significant. Then the result in this study, it was found that BMD of all skeletal site was significantly decreased while BMI years is menopause, and weight was significantly higher in postmenopausal osteoporotic women when compared with healthy women. The genotype frequency were cases the wild type homozygous 36.6%, heterozygous 51.6%, and mutant is 11.8%. And the control genotype frequency were homozygous homozygous wild type 29.9%, heterozygous
You may conclude, ma'am.
59.9%. Hello.
Time's up. You may conclude, please.
Okay. So the result was showed that higher BMI or menopause were found to be independent significant predictor of osteoporosis, but results were not significant difference for allele and genotype in control and patients.
Thank you, ma'am.
Ma'am, can you explain why we need to study association between the osteocalcin and the bone density, ma'am? What is the importance of studying this association?
Sir, actually, Doi et al. Identified a polymorphism of osteocalcin gene within third restriction enzyme, but our result were not so significant in North Indian postmenopausal women, means negative result.
Okay. What do you want to suggest to the society with this finding? Do you have any message for the society?
Hello?
Like if you want to create awareness about something after finding something, what do you want to convey to the society?
So your voice is not clear.
No. Do you have any suggestion for your target group of people? Do you have any suggestion for leading a healthy life?
Sorry, sir. Your voice is not clear.
It's okay, ma'am. I will send a text message about for your question. You can answer it by sending a text message back. Thank you.
Thank you, ma'am. Next we call Ghazala Shaheen. Slot number 3.
Okay, thank you, ma'am.
Yeah, thank you, Ms. Priya. Slot number 4, Monica Jaitar, please get ready. Ms. Ghazala Shaheen, are you in line?
Yes, ma'am.
You may present now.
Yes, ma'am.
Ms. Priya, can you stop sharing your screen, please?
Yes, ma'am, I'm sharing. Ma'am, can you please close the previous screen?
Ms. Priya, are you in line?
Ma'am, the other screen is sharing, that's why I'm unable.
Yes, once again. Okay, now you may share, ma'am.
Yes, ma'am. Ma'am, can you share my screen? I have shared it in the WhatsApp group.
You are not able to share now? Aren't you able to share now?
Ma'am, I'm not able to share it. Can you please?
Okay, ma'am. You just wait for 5 minutes. We will be sharing for you. Please be online, okay? We will call you next.
Okay, ma'am. Thank you.
Yeah, thank you. Monica Jaitar, slot number 4. Are you ready with your presentation? Miss Monica Jaitar.
Yes, ma'am, I'm ready.
Please present, ma'am.
Okay, ma'am, just give me a second. Can I share my poster, ma'am?
Ma'am, then able to hear you.
Listen, mic on echo.
You may start, ma'am. We can see your poster. Can you just make it in full view? Yes, thank you. Start.
Give me a second, ma'am.
You can start, ma'am. It's fine. We are able to view it.
Am I audible?
You may start now. You're audible.
Good afternoon. Hello? Ma'am, is I audible?
Yes, you're audible. You're audible. You just start, ma'am.
Hello?
You're audible, ma'am.
Ma'am,
You
shall
can
I start?
You can start.
Good afternoon to all of you. Today, I'm Monica, a research scholar from University of Jammu, is going to present on the topic Association Analysis of HNF1 variant with MODI in population of Jammu region. MODI stands for Maturity Onset Diabetes Young, is caused by single gene mutation that lead to defect in beta cell insulin secretion in response to glucose stimulation. There are 14 gene mutations which are present. One of them is HNF1A, which stands for hepatocytes nucleofactor 1 alpha. The HNF1 is expressed in pancreatic beta cell, the liver and intestine, and it is critical transcription factor for insulin gene and GLUT2 in mature beta cells. HNF1 mutation are the most common cause of MODI in Europe, North America, and Asia. The most commonly reported mutations are missense mutations. It is slowly progressing and predicted to be linked with T2DM. This gene has been inconsistently associated with increased risk of T2DM. The missense variant within HNF1A is found to be associated with MODI in various populations worldwide, including Turkish, South African, Sudan, etc. So aim of this study is to investigate the association of RS11692888 with MODI in population of Jammu region. As there are 14 different genes and MODI is the most common form of monogenic diabetes, patient type is caused by affecting beta cell insertion in its inherently autosomal domain. These four different genes like GX1, INK, HNF1 alpha, PAX4, ABCC, and J11, Neuro D1, KLF11, and CEL. This is the autosomal dominant inheritance pedigree, and this is how autosomal inheritance is passed on to offspring by using bioinformatics or bioinformatic tools we try to, this is the location of the SNP, and we use the string database to provide the protein-protein interaction, that is HNF1A alpha with HNF1 beta. Ma'am, I'm audible now? Hello?
Yes, you are audible.
Hello? Yes, ma'am. This is the gene location. Material methods are used.
Your time is up, Monica. You may conclude now.
Sorry, ma'am.
Your time is up. You may conclude now.
Okay, fine. So, with conclusion, I would like to conclude with that HNF1 gene, the SNP of this is showed polymorphism and allele T allele with prevalent in Turkish population, but it was not significant in North Indian regions. So these are references, and these are the PCRs and heterozygotes. Thank you.
Thank you for your nice poster, ma'am. Have you arrived at
Hello. Hello.
Have you arrived at allelic frequency of the
You are not audible. So can you just repeat?
Have you arrived at allelic frequency of general population?
Different population.
Yeah.
Not
yet.
Not yet.
Okay. Thank you.
Monica, you may stop sharing now.
Hello.
Slot number 5, Mahalakshmi. Slot number 5, Mahalakshmi, are you there? Slot number 5, Mahalakshmi. Okay, slot number 6, Sri Sarika, are you there?
Yes, ma'am. Am I audible?
Yeah, you may share now.
Okay. My screen is visible, ma'am?
Yes,
Hello?
Sarika, it's visible.
Okay. Thank you. Good afternoon, one and all here. Myself, Sri Sarika, studying biotechnology at Ratnam College of Arts and Science. My topic is about green synthesis and characterization of silver nanoparticle using cis-alphinia bonded plant extract and their antimicrobial properties. Okay. First of all, recent advantages in nanoscience and nanotechnology changes the way of diagnostics. Using silver nanoparticle for treating disease is more effective. Compared to the other metallic nanoparticles, silver nanoparticle is most effective in treating disease. So we take the silver nanoparticle as our study, and our plant sample is cis-alphinia bonded from Fabaceae family. It also has such kind of properties like abscesses and as well as anti-diabetic properties and wound healing properties. So we take the cis-alphinia bonded plant extract. We go for the synthesization after adding 1 mm of AgNO3. The silver nanoparticle will be synthesized in 24 to 48 hours. As you see, after 24 to 48 hours, the plant sample changes the color in brown. So that we're confirming that the silver nanoparticle were synthesized, and also we go for the characterization study in UV spectrometer. In the UV spectrometer analysis, our result of expecting silver nanoparticle is 446 nanometers. So we confirm that silver nanoparticles, and we also still studying in scanning electron microscopy and transmission electron microscopy for characterization. It is still in work, and we also going for the antimicrobial activities. In those activities, we using E. coli and Pseudomonas. In our plant sample showing zone formation about 15 millimeter in E. coli and 20 millimeter in Pseudomonas aeruginosa. So our plant sample is showing good antimicrobial properties, and we also currently working on anti-diabetic and wound healing properties. So our conclusion is it has some good anti-diabetic properties and wound healing properties. So we going to study in wound healing for diabetic patients. So we recommend it for wound healing study, and we also ongoing study about the wound healing properties. So this is my presentation. Thank you. Hello.
Yeah, thank you, Sarika.
May I know why you have chosen this Cisalphinia bonded plant?
Sir, our study is mainly to identify anti-diabetic properties and wound healing properties. So our plant Cisalphinia bondic has such kind of properties. So we can apply this properties to wound healing for specifically for diabetic patients. So we take that Cisalphinia bondic plant, sir.
Okay, what could be the advantage when you compare the already existing literature about wound healing property of a particular plant? What is the advantage of preparing nanoparticles and then treating the same wound?
Sir, as I told, sorry.
Yeah, please go on.
It is an effective way to treat the disease. Maybe nanoparticle synthesization, nanoparticle based medicine can cure easily than other medicines. If we are intaking the solid type of medicine, means it will be it may be slow, but our study nanoparticle may cure the disease easily. So we using those methods.
Okay, thank you. We can move on to the next presentation. You can close your screen.
Okay, thank you, sir.
Slot number 7, Pavitra. Slot number 7, Pavitra, are you there? Slot number 7, Pavitra, are you there? Slot number 8, Sumisha. Slot number 8, Sumisha. Slot number 8, Sumisha, are you there? Slot number 9, Anushri Devi, are you there? Slot number 9, Anushri Devi, are you there? Slot number 10, Shanmugapriya. Slot number 10, Shanmugapriya. Slot number 11, Rajalakshmi. Slot number 11, Rajalakshmi. Slot number 12, Soumya.
Yes, ma'am. I'm ready, ma'am. Soumya will be ready, ma'am. Can you hear me, ma'am?
No, sir. Actually, illa sir. Avanga vera PNJ irukanga. Yeah, their ID is actually different, so I couldn't find the ID.
Soumya, are you there?
Yes, ma'am.
Yeah, you may present.
Can you hear me, ma'am?
Yes, you're audible.
Can you see my screen, ma'am? Can you see?
Yes, it's visible.
Shall I start, ma'am? Good afternoon, we are gathered here. My name is Soumya. I'm a research scholar doing my research under the guidance of Dr. R. Jasmine, ma'am, Department of Biotechnology, Bishop Heber College. Today, I'm going to present one part of my work, that is metagenomic analysis of the gut microbiome of Callus domesticus and detection of allocytes in distinctions targeted against colorectal cancer. Introduction. The road to health is paved with good intestines, said by Sherry Rogers. Nowadays, everybody is looking for alternative for antibiotics. So this is one of the boom topic, that is microbiome as a biomarker for cancer. So this gut microbiota derived metabolites are key molecular mediators between the host and the microbiota. So what is the objectives of my study is to isolate the probiotic bacteria from the gut microbiome of Gallus domesticus, that is chicken, broiler chicken, to identify and characterize the isolated bacteria and obtain and screening the metabolites from the isolated probiotic bacteria. Finally, GCMS analysis to identifying the compounds and to evaluate the metabolites activity in silico and in vivo. And this is a methodology what I have followed. That is the sample collection from the chicken gut and the isolation of probiotic bacteria and the screening and characterization of probiotic bacteria and the screening for the metabolites, that is lipid, and bioassay anti-cancer activity, that is the initial screening activity, that is MTT assay, and GCMS analysis and characterization by 16 years rRNA amplification. This is the results. From the gut, I have isolated one probiotic bacteria with the help of molecular characterization. I have found that it will be Elistypes indistinctions and it will be submitted in NCBI. And this is the NCBI. This is 1365 base pairs will be there. And I have characterized these bacteria. It will be a gram-negative anaerobic non-spore, and it is a urease, nitrogen reductase negative and catalase positive, and the metabolite isolated from this is a lipid, and this is followed the standard protocol. This is the characterization of the particular bacteria, and this is a lipid sample. Finally, I have gone for this lipid sample for GCMS analysis. I got more than 30 compounds. In among that, I have chosen a top 4. In among that, with the help of literature study, I have chosen the hexadeconic acid methyl ester for in silico analysis. At the same time, initial screening for anti-cancer MTT assay performed. Here, this is a control cell line and this is a treated cell line with my metabolites. I got a predominant results from this. And molecular docking, I have chosen, so the IC50 value of tested sample is 163.6 μg per ml. And I have gone for the in silico analysis. I have chosen the beta-catenin protein. And this is the initial protein which involving in the process of colorectal cancer. So I have chosen this protein. And this is my compound. When while docking study, it's showing a very good binding energy. So the binding energy will be minus 6.37. So more than 30 the conclusion, more than 30 bioactive compounds identified. In among that, I have chosen one particular compound showing a very good binding affinity towards beta-catenin protein. I have done this docking study. Yes, ma'am. Yes, ma'am. So in the conclusion, in my research, I have found the lipids and isolated the metabolites which produced from the Elistypes intestine, shown a very good activity against the colorectal cancer. That's all about my presentation. Thank you for giving me this wonderful opportunity for the organization. Thank you so much, ma'am. And if you have any questions, please ask me, sir.
The compound which you have isolated is from probiotic bacteria, right? Is it a probiotic product?
Sir, can you please tell me again, sir?
The compound which you have isolated and suggesting to treat the colorectal cancer, is it a product of bacteria, right?
Yes, sir. It's a product of bacteria. It is a metabolites. The compound I didn't exactly isolate, sir. This is the metabolites. In the metabolites only I have checked the activity.
So have you standardized the culture conditions for this bacteria?
Yes, sir. That is a screening I have done, sir. The characterization, it is a characterization test. It's a catalase and urease and bile salt tolerance. All the characterization I have done, sir.
Okay. Okay. What are your future plans with this work? What is your future plan with this work?
So nowadays everybody is using a lot of antibiotics. So without looking outside, from our gut microbiome itself, if we enhances the gut microbiome with the proper diet, means our gut microbiome will produce a metabolites that will help to increase our immune system. So this is one part of my study only. I have treated that particular chicken with some I have treated the chicken with some supplementation, sir.
Okay. Is it possible to prepare edible capsules with this bacteria?
So can you please repeat?
Nowadays, lot of prebiotic capsules are available.
Okay.
Is it possible to prepare capsules, edible capsules of this bacteria?
So this is you are asking on the future edible capsule or now available?
Have you done any study about safety of using this bacteria as an agent?
Sir, just now I am just clearing the initial screening, so I am not targeting in the capsules. And also, once we are giving some modulation of the diet, means automatically our gut microbiome will produce the good bacteria. So that good bacteria will produce metabolites. Automatically, that will increase the T cell receptor. This alloceptase indistinctions will major role is T cell differentiation. That is T cell receptor. So it will give the immune response. We will get the strong immune response.
Okay.
Then automatically the initial step of the colorectal cancers will be prohibited, sir.
Okay, thank you. Thank you.
Thank you.
You can switch off your screen so that other person can start the presentation. Thank you very much. All the best.
Thank you, sir. Thank you so much, ma'am.
Slot number 7, Pavitra. Are you there? Slot number 7, Pavitra. Slot number 7, Pavitra. Slot number 13, Yamuna.
Yes, ma'am. Yes, ma'am.
Yeah, you may proceed now.
Is it audible, ma'am? Ma'am, am I audible?
Yes, Yamuna, you're audible.
Okay. Topic Ma'am, is my screen visible?
Yes, Pavitra, you're visible.
Okay, ma'am.
Could you please zoom out? Pavitra?
Yes, ma'am. Yes, ma'am. Yes.
Yeah.
Is it okay now, ma'am?
Bit more. Yes, fine. Yeah. Okay.
Good afternoon to all. My name is E. Bavitra. I'm first year BSc Microbiology, Kamaraj Women's College, Thuthukudi. My topic is Translate Schema from Batch 2, Big Science and RRTs. What is translation research? The goal of translation research is to the translate-based science discovered more quickly and efficiently into practice, applicable for the betterment of society. The importance of translation research and the collaboration among laboratory and clinical research and determine general public their needs for health innovation. And 5 phases of translation research, T0 to T4. T0, basic science research, T1, translation to human, and T2, translation to practice, T3, translation to patient, and T4, translation to community. Population level outcome research, the true benefit for society for human health. Rare respiratory disease, LAM, COP, HPS, PhD for the rare respiratory disease, and the traditional research. Rare lung disease. Recently affected the SARS-CoV virus, direct effect to the lung, pulmonary edema, dyspnea, acute respiratory disease, acute lung injury. The small part of alveoli, the image of the type 2 pneumocyte cells and the stone, cytokine stone, the direct effect to the liver and kidney. Leukopenia, thrombocytopenia, lymphocytopenia, then direct effect to the blood cells. And systematic and respiratory disorders. The COVID affect man, the systematic disease is the fever, headache, and dry growth. The respiratory disease is the nuclear and breathing. Thank you, ma'am. Thank you for giving me the opportunity.
Thank you. Do you have any references for your work? If this is a review work, do you have any reference?
Yeah, we have given over here, sir.
Have you collected this from single article or several articles?
We have collected from several articles from biomedical journals, research journals. Since we are from first years, we have collected we have done the review articles presentation.
Okay, thank you. We can move on to the next presentation. You may close your screen now.
Thank you, sir.
Thank you.
Thank you.
Slot number 13, Yamuna. Slot number 13, Yamuna.
Can I share my screen, ma'am?
Yeah, sure.
Okay, ma'am. You visible my screen, ma'am?
Yes, Yamuna, you're visible.
Okay, ma'am, fine. So good afternoon to all. This is Yamna, you a student in Department of Biochemistry in Rabi Mohamed Maidin College for Women, Tiruvarur. Today, I gonna use regenerative drug which was formulated by me. Why I propose this drug means, as we all know, nervous system has a complex and delicate structure. Patients with serious nerve disorders like Alzheimer's disease, Parkinson's disease, they often require lifelong assistance. So to nourish our nerve and to cure nerve regenerative disorders, you can take this drug to heal it. So you can see this image. This is the image I propose the drug. It not only cure nerve degenerative disorders, but it also cure insomnia, severe acute demyelinating lesions, polyneuropathy, acute disseminating encephalomyelitis, and dysautonomia. These are all the disorders caused by coronavirus on neuron. So you take this, when you take this drug, it will cure by nourish your nervous system. So now we're gonna see phytochemicals present in this drug. First one will be the vithnoside 4. It will metabolize into somnion and it will help to recover the nutrients and synapses. And within somniferin A has reduced the and it reduces anti-angiogenic influence and promote angiogenesis. It is essential for nerve regeneration. It protects against stress-induced gastric ulcer. This help also modulate brain oxidative stress makers and neurotoxic makers. Next one will be the rutin. Rutin has an antioxidant effect, anti-apoptotic effect, and anti-inflammatory effect. And next one will be the ascorbic acid. It is essential for development and physiological function of nervous tissues. And next one will be the piperine. It has a pleiotropic properties because it acts as an anti-elliptic, antioxidant, antidepressant, and it also helps to treat sciatica and it reduces oxidative stress. Next one will be the phytosterol. I obtained this phytosterol from Solanum trilobatum plant. So you know what? If a people expose long time in a pollution, lead molecule from the pollution.
Now?
Okay, ma'am.
Yeah.
So I conclude this topic. So when you take this drug, it slowly get nourishment to your nervous system and it will cure nerve regenerative disorders and then your nerves get regenerated. Thank you, ma'am.
Have you analyzed any natural product that can regenerate nerve cells?
So these are the phytochemicals I have given here. So these are all have the ability to cure by nourish the nervous system, sir.
Okay. What are all the sources? Are they indigenous plants or exotic ones? Any idea?
Sir, I can't able to clear.
I mean, I wanted to throw light. I wanted you to throw light on which of these plants are from India or which are the herbs from abroad, like exotic plants.
Sir, these are all plants obtained from our place in Tamil Nadu. I have taken 7 plants, sir.
Okay.
Okay, sir.
And do you have any idea about synthetic chemical drugs that are having therapeutic properties regarding nerve regeneration?
If you take a synthetic sense, it will, when you take this drugs, it will cure, but it does not produce any side effects, sir. So when you take this drug, it is.
It's
Sir?
good that you have screened so many plans, but my little suggestion is you have to for validating your study, you have to compete with some existing drug. Only then you can validate your product. Okay.
Okay, sir.
Maybe in your future studies you can compare with the existing drugs. Okay, thank you.
Okay, okay, sir. Thank
You
you,
may
sir.
stop sharing your screen, then we will move on to the next one.
Okay, sir.
Here we have our Sir Dr. P. Suresh Kumar, sir, the Professor and Head of Department of Biotechnology from Anna University. We welcome you, sir. Good evening. So now we move to the next person. Miss Monica Jaital, are you there in line? Slot number 4, Miss Monica Jaital. Okay. Anushri Devi, slot number 9, are you in line?
Yes, ma'am.
Please present.
Yes.
Yes, you may start now, ma'am.
Yes, ma'am. Yes, ma'am. Just a moment. Good evening, ma'am. I'm Anushri Devi of 2nd BSc Microbiology from Kamaraj College. My topic is on the recent catapulted progression in nanotechnology against the treatment of respiratory disorders. First of all, I'm going to tell about nanotechnology. Nanotechnology is the manipulation and reduction of materials at the nano size, that is 1 to 100 nanometer, with distinctive characteristics and properties. It has a range of applications, and one among them is the nanomedicines. The nanomedicines ranges from the medical applications and nanomaterials to nanoelectronic biosensor. Current problems in the nanomedicine involve the understanding the issues related to toxicity and environmental impact of nanoscale materials. Now let's have a glance on the sources of nanomedicine. Major sources are from the natural biopolymers extracted from microbes, algae, and higher plants and animals. The structure and size of nanomedicines ranges from dendrimers 1 to 10 nanometer to liposomes and nanosuspension 30 nanometer to several micrometer. The types of the nanomedicines includes the polymer-based nanoparticles, example micelle dendrimer, inorganic or metallic nanoparticles such as quantum dots, silver nanoparticles, lipid-based nanoparticles, and protein-based nanoparticles. Now we could see the golden age in the development of nanomedicines was from 2010 to 2011. The efficacy of nanomedicine depends on their systemic absorption. It differs with each and every patient. It is applied for the betterment of the society. The clinical translation in this, the rational design was created, and the high throughput screening was performed in mice and monkey. In case of the positive result, clinical trials were performed. Then the scalable production of the nanomedicine is carried out, and finally it is marketed in the markets for the betterment of the society. Now let's have a look on the pathology of COVID-19. The coronavirus genome is a single stranded RNA having diameter of 60 to 140 nanometer, and so it is easily transmitted through airborne and respiratory droplets. That's why the nanomasks such as N95 are highly recommended. The primary organ that is directly affected by COVID-19 is the lungs. The delivery of drug to the lungs is compelling because of its significant systematic and local effects. The nanosensor using quantum dots, carbon nanotubes, and metal nanoparticles are used in the diagnosis of COVID-19. Protein corona sensor arrays, magnetic levitations are used in diagnosing high-risk patients. The cycle of COVID-19 within the host body involves 5 steps: attachment, penetration, biosynthesis, maturation, and release of the virus. And it causes cytokine storm in ARDS, that is acute respiratory distress syndrome. The treatment of this include the small molecule drugs, exosomes, immune regulation, and nanovaccines. The nanomedicine has a great ability to directly deliver the drug to the lungs, especially in ARDS.
Manushree, the time is up. You may conclude now.
Thus, I would like to conclude that the nanomedicine has a great efficacy in treating these respiratory diseases. And thank you for giving this opportunity.
Thank you, ma'am. Do you have any examples for nanomedicine that is available in market? Any idea? Any example can I get?
Sir, I have not reviewed that.
Okay, you have done a nice poster. Better if you go for live examples, like what is the currently existing technology. Better add more value to your poster. Okay.
Covaxin and COVID.
Okay. Thank you. You can close your screen and we will move on to the next presentation.
Okay, sir. Thank you, sir.
Slot number 10, Shanmugapriya. Slot number 10, Shanmugapriya.
Ma'am, I'm there.
You may proceed now.
Lung injury. Share.
Good afternoon, everyone. I am Vidya, second BSc Microbiology from Kamraj College. I am presenting for my batch. My topic is an obvious light on certain combinatorial therapeutic schema to cure human lung injuries caused by the coronavirus. Nowadays, there are various therapeutic approaches are available for the treatment of COVID-19. It includes serum therapy, immunotherapy, nanomedicines, etc. Next, we could see the lung injuries caused by the COVID-19 infections. What is lung injury? When a person is affected by coronavirus, his lung is injured, so there is vasodilation and increased capillary permeability, resulting in cytokine storm. Due to the lung injuries caused by the COVID attack, the entire oxygen transport will also be affected. It results in the entire systematic damages and increases the activity of our immune system, which are described below. Moreover, due to this drastic inflammations in the respiration system, the entire body of host gets collapsed easily. In vessels, inflammation into the entire cell layer and lymph vessels. In heart, myocarditis, arterial inflammation. In kidney, total kidney was damaged. In lungs, lungs are stiffened and irregular breathing. In brain and nervous system, we could see the taste and smell loss. In other organs, liver damage and muscle pain. Nowadays, with the aid of combinatorial approaches are applicable for the recovery of this pandemic, such as acupuncture and yoga, which boost our immunity and strengthen lungs. Thank you. Everyone should join hands to recovery from this disease. Thank you for giving this opportunity.
Thank you. You can stop sharing your screen. We will move on to the next presentation.
Slot number 11, Rajalakshmi. You may present now.
Biotechnology.
Yes, ma'am, I'm here.
Poster presentation
Yeah.
poster presentation இவங்க முடிக்கட்டும் அதுக்கப்புறம் from the next slot
we give the separate link for this poster presentation. Now the validity session is going to start. So it is Suresh Kumar sir. Call
Just hold on. We'll give another link and then ask them to download.
Just a minute. Just a minute. Hold on, Rajlakshmi.
Ma'am, may I start the presentation? Am I audible, ma'am? Ma'am? Excuse me, ma'am. Am I audible?
Hold a second, Rajlakshmi.
Yes, ma'am.
You may end the presentation. We'll send you another link. You may join in that.
We have to join another link, ma'am.
Yes, yes, Rajlakshmi.
Okay, ma'am. Will it be shared in the WhatsApp group?
Yeah, they will send you in WhatsApp group. You may join in that. Okay.
Okay, ma'am. Okay, ma'am.
Yeah.
Thank you, ma'am. Okay, stop share.
Thank you.
Yes, ma'am.
All the participants may join in the other link, which will be sent in WhatsApp group. All the participants may join in the another link, which will be sent in WhatsApp group.
Ma'am, shall I leave this group, ma'am?
Yes, yes. You may all leave now. You may all leave.
Thank you, ma'am. Thank you.
Only post the presentation. The remaining participants on online can stay for primary lecture 6. Once again, I repeat, the remaining online participants may stay here for the next daily lecture. Only the poster presentation participants may leave. Thank you.
Just wait me. I present.
Yes.
तेरा ही चांस
Good evening to one and all gathered here. It gives me an immense pleasure to introduce the chief guest of today's program, Dr. P. Suresh Kumar sir, Professor and Head Department of Biotechnology, University College of Engineering, BIT Campus, Tiruchirappalli, Tamil Nadu. Sir has nearly 20 years of teaching experience and have been in the field of research. He started his career as an executive with Sartorius Biotech Limited, India Limited, Bangalore, in the year of 1996 to 1997. Sir has specific area of specialization in research includes transgenic technology enhancement and production of phytochemical and nutraceuticals, drug discovery and development, phytodrugs, green synthesis, and bioactivity study of nanoparticles. Sir is a recipient of many prestigious awards like Young Talent Awards 2016 given by Association of
Pharmacy Professional, Madhya Pradesh, India. And also awarded as the Young Scientist Award, Department of Science and Technology, Government of India in the year of 2008. Senior Research Fellowship, Indo-Asia Collaboration Project, Department of Biotechnology, Bharat University, Tiruchirappalli in the year of 1999 to 2000. Said also being a membership of Scientific and Professional Society like Indian Science Congress, European Federation of Biotechnology, Indian Biophysical Society. Such an eminent person is with us. I'm really proud to introduce him. He has published more than 30 papers in most reputed international and national journals and has completed many research-funded projects from GBT, DST, SCRC, DST, SCRD for the worth of 1 crore. Such a dynamic and eminent person with us. Once again, I welcome you, sir. Thank you. Now, I kindly request Dr. P. Suresh Kumar sir to lecture the gathering. Please, sir.
Screen share start.
Good evening, all of you. Thank you. Yeah, I'm happy to be here. The third time I'm visiting your campus. Actually, I was there invited by the principal for project graduation. Some of the faculty could remember me. And I was also in your staff selection. This is the first time I'm coming to the institute to deliver a lecture. Normally, my expertise was used only in selection and screening of projects. And I asked Benazir how she picked me up from internet. Of course, I have some of my friends over here, but she said that she picked me. Oh, I asked her immediately how were you able to identify me? She said, sir, I saw your profile on the internet and then I checked you from there. Surprising, but anyway, internet gives you a lot of things, right? The positive things you can search. We will all doing research in that area. Of course, honestly speaking, I'm not a researcher in COVID-19. Before starting, I'll tell you, my research area is in diabetes and more emphasized on phytodrugs. So when I asked Benazir what to talk, because I'm more expert in the COVID complications or of course, I know certain aspects, but I'm no expert in that. I said, fine, sir, you talk about your research experience that will be more useful to our students. And I asked about the audience, he said about BSc and MSc students or their research scholars. I'm happy that to speak with the young minds, dynamic minds. Yeah, I'm almost thrilled, like as a teacher for years of 2 decades. More I like to address the students or interact with the students since I'm handling my BTech Biotechnology and tech students there. So no wonder I'm just carried over to your institute. The student is a student, whether it's a BTech or a BSc or MSc is all fine. And biotechnology is so interdisciplinary that all disciplines of research which comes under biotechnology have no gradations or no discrimination. Okay, research is a research and publication is a publication. Okay, that's one of the things. So we will start with that. This is my institute. I know it's very difficult to sit from the morning to evening, especially the last peg end of your program. You go to receive certificates and so curious to know about your poster presentation. You are going to build that, like in TPM Max, right? So I don't want to take much time. I'll take half an hour or 45 minutes. So the choice is mine or theirs?
Yes,
I think I will not bore you. Until I asked, I was so frustrated and reluctant to what to talk to you because I can see all the MSc, just you're exposing to research.
I'd like to share some of my expertise. It's rather good to talk about what I did rather than what is there in the net. Got me point? So that would be more interesting than what I did. You want me to talk in bilingual or also to that? Or you just find it bilingual?
Yes, sir.
Huh? Good. So this is my institute. You can see the text of course is my entrance, and then this is the entrance view of my institute. Some of you have crossed to the hotel before here more my institute. Started in 99 with Harvard University and subsequently now anniversary. And you have all the liberty to stop me if you don't understand, okay? You feel that I'm out of focus. Okay? You don't understand, you have to stop me. Okay? So you are at liberty now. So the emphasis is that when I saw the theme of your conference, it spoke about the COVID complications, post-COVID complications. We all know that one thing that even now the admission on biotechnology is moving out relatively all lives. You know why? Why the interest now focus? The entire world is now focused on biotechnology research or biological research.
Find the solution of the serious problem.
Yeah, so see the one thing that is in technology or artificial intelligence, you may make a missile, you can have substituted 5G now 6G. Telecommunications, electronic communications, advancement, military advancement, and other scientific networks. That has the entire 2 years of past COVID. You have been locked down, right? So simple miniature microscopic virus put us down at home, right? That is what this the entire world has hit the panic button. No importance of the biological research. Got my point? Why the craze and now again the biotechnology or the biological research is gaining momentum because of this thing. If more people or even the engineering people are more interested about how COVID has the mechanism of COVID, how to control, what are the symptoms, how to avoid that. So all these things comes, whether it's a non-biology or engineering, whatever stuff, they're all more focused. Everybody knows about coronavirus, right? That is the beauty of research. Okay. So I'll just quickly go through my slide. Okay. Next. So primarily we talk about the drug discovery and development. That is again the impetus now. The entire world is focusing on the drug development. So somehow we have to find some vaccine for this virus, virus. Even after vaccination, people are getting infected. Why is it? Why? And what is the process involved in the drug discovery? So people are more focused on now. People even ask any common layman, they talk about the clinical trials. The vaccine has been drug developed. Vaccine, clinical trials. All these things are now become a common words. Earlier, it was not only biological student or researcher. So that has given an impetus that the focus on how the drug is discovered. Just take any medicine, whatever medicines you buy from your shop, medical shop. It takes around 13 to 14 years of research and the surveillance, FDA approval, all this stuff to really get a single drug into the market. That's the reason, even after 2 years, they are able to find a vaccine. How effective the vaccine is. That's why they are probably more the protocols of the drug development. Now, when I saw my slide, sometimes I mean that within 2 to 3 years, we are able to find a vaccine. This is again because we have further go, find an alternate, find a methods to control the COVID or the coronavirus infection. And that is again a concerted effort of throughout all sciences of thought, because it needs a profound effort. And the virus infects the type of virus is mutant. The virus, they are very dynamic. And it can keep on changing stage, size, and infection process. It's very difficult to control even the cold virus. Yeah. 3 days you have to take a medication, antibiotic, after something. So, not imagine that every virus like COVID. So situations are there to how to minimize within the thing. Otherwise, we cannot put people locked down for years together and bring down the economy of this world. We have to come out and work. So we are not going to have a production of a vaccine or a production of a drug within a short period of time. But with all the high throughput technologies available, scientists throughout the world focus, and then we have produced some vaccines. And you know, the entire world has looked upon us to produce a vaccine. The Secretary of WHO has said that we look upon India. Why? We are the one who produces vaccine which can eradicate smallpox and polio. India has a tremendous potential of controlling threats. It has teach the entire world that how to vaccinate people. So we may have some problems over there with socio-economic problems, but anyway, India is one of the leaders in the vaccine production and on implementation of vaccination among people. But the entire world looked upon us for a vaccine production. So this is the story, okay? Next, we talk about technically how to produce our drug. Is that I told you it is not only a biotechnology. I told you research is nothing but an interdisciplinary approach. Whatever research you do, it is not concerned only with one particular domain. Don't restrict yourself. It may involve to say like from pharmacy, biology, chemistry. Even you need a bioinformatics, a computer specialist, manufactured drug, you need a reactor, you need a mechanical reactor, bioreactor. We need the communication people to link all the sensor, and we need a computer person to program to on-off control. The big reactor we cannot manually switch on to control, but we need an on-off control system which is learning program. We have program logic control. So like that, we need a concerted effort. If the producer drug is not only a single man, single discipline, it needs involvement. After that, the drug has to be marketed. It has to first be prescribed the disease in your doctor. It has been marketed through your pharmacy salesman, and it has to reach the public, isn't it? Just see from lab to plant and then to the market. All these steps is involved. How many papers are involved? It's not claimed that research is I did. We did. So never use the word I did. I did research. It's not the I did. It is again is a team. So we can see that there were new potential therapies were identified in terms of the modern drug discovery. Earlier drugs were discovered to known active compound was called lead compound. I'll show you what I did. Okay, next slide. And actually, my research is on how I identify a compound. It is called the lead compound. It's supposed to be a drug. How I extracted from the medicine, how I identified the medicine plants, how I executed, how I extracted from the phytodrugs, and how I tested, and how I promoted as a lead compound. So my work stops up to lead compound. Rest of the industry aspects, the pharmaceutical company has to take over from there for a clinical trial. What I want? Two phases of research. Up to preclinical, after clinical trials, and FDA approval. There is another process. So the most importantly, we should understand before identifying the pathophysiological conditions at the molecular level of diagnosis of the drug. That is again very much prerequisite first for a drug discovery. Next. So when you really are looking for a drug discovery, it's a new target. How to find a new target, identify a drug target. It is PPR gamma. PPR gamma is a receptor for diabetic diabetes. Okay. Okay. So since I work on diabetes, so I have a specific target. A receptor, diabetic receptor, which will transfer the glucose. Okay. Simple, I put it in that. That's what my research is there. And you would have to do some biological assays, both in vitro within the laboratory using the biochemical assays, and in vivo using animal models. And then later you can go for the toxicological studies. This is how the process takes before clinical. And drug development, after that, when you identify the lead compound, it has to go for a pharmaceutical scale up. How to scale up the compound in large scale, industry scale up. Whether you use bioreactor, whether you use chemical synthesis, or you can use any kind of synthetic synthesis of your lead compound. That's again is very important step. Then you have the ADME, essentially absorption distribution studies, to test the toxicological effects of your compound that you identify as drug. So you have to do toxicological studies, and then now you formulate. Once you say that the toxicological studies are very fine, then you go for the formulation as a medicine, as a drug. The drug means it may be a small microgram level. You have to coat the drug in a tablet form, so it has a coating. Then either in the form of coating or injection or in terms of antiseptic cream or whatever it may be, any topical, it all depends upon the mode of delivery of the drug. Okay, and that's called the formulations. Then you call clinical trials. It's a story that India has been like a guinea pig anyone. Okay, like guinea pig, what is guinea pig? We are the testing ground for all the Western medicine. Gujarat, there was a famous saying that people have been paid 4 lakhs, 5 lakhs, and then they give a drug, but they reduce the disease. They treat the patient with the drug, not approved drug, the drug which has to be approved by PA. They test with small number of patients from 30 to 100, 100 to 3,000, 1,000 to 3,000, like that. This is how the clinical phase 1 to 3. So when you identify whether because of the drug is there any side effects or so if there are any side effects or there no side effects, then they'll approve the drug for FDA. Okay, that's a process. Okay, so when you this every clinical trial will take 1 or 2 years. Imagine for 3 clinical trials it takes 6 years. We have released the drug within 2 years. Okay. So drug elucidation is again either by using model. This is here again. Another one is we use mice model now. Rat. Sometimes when I worked in Mumbai, our pharmaceutical company, before we joined university, I saw surprisingly we use also horse and dogs for testing. Then the pharmacological intervention, like you see, when you put a drug in your body, your drug dynamics, we call pharmacokinetics or pharmacodynamics, how the drug is getting active in your body, how your body is responding to the drug. That's very hard to do. Then the behaviors because of the drug is that any psychological changes. Some drug, if you give on take, you have a psychological disorder. When you go into depression, what will you want about? I continuously in days of the especially the chronic disease like cancer and diabetes. Chronic intake of chemotherapy in cancer patients is so better that the side effects are more than actual cure. So the point here is emphasized is that how to reduce all these side effects. We cannot say that any drug is safe. Whether it's a bio drug, meaning whether it is a medicinal plant drug or it's a synthetic drug. All drug has its own adverse reaction, adverse side effects. Or when we identify that and we rectify that, that is the essence of research. Next. So I told you, like if you screen around 5,000 to 10,000 compounds, that is called the target identifications. Then you, after the assay development and the lead generation, you may end up into the compounds. That is the preclinical. Preclinical is what I talked about using the rat model, rat or mice model, using animal model. Okay, cell lines or animal model. Cell lines comes at the individual model. In vivo model means animal models. Then after that, you end up with 5 compounds for clinical drugs. These 5 compounds are in the regulatory affair, FDA, Food and Drug Administration of USA, will approve one drug. So you just see how much of phases is involved. You can see around 15 years. Because it needs, because when you talk about the human health, immediately a drug cannot be released in the market within FDA. So to get an FDA approval, this is the process. For any drug, even the market, the drug you are currently consuming or getting it from the medical shop, requires this much of effort. Next. This is what I told you. It starts from 20 to 80 participants to 100 to 300 and 1000 to 3000. And then after that, to confirm safety and effectiveness for FDA review, and then the report that drug is approved. We again got post surveillance marketing. That means after the release of approval of the FDA, after 1 year, is that any patients or have encountered any kind of side effects or any adverse effects of consuming the drug? You may never know. When I did 2010, I wrote a project, the drug which is commonly used for diabetes is oxyglutazone and 5. Now we know diabetes is very common household name, like COVID. In a family, at least one of you got diabetes. Especially when you grow old, whether the parent has inherited you some wealth or not, definitely some cases. Yeah. So diabetes is again inherited. The people said if your father and mother diabetic, you are prone to get diabetes because of genetic predisposition. Okay, so that again is the story that we talk about. But we have to be very careful with our diet. We have some other things to prolong the diabetic complications or diabetes. Okay, so after continuously taking this drug, I'm telling my story about I have worked on oxyglutazone and 5. These were 2 drugs which being used for and the anti-diabetic drug. Okay. It has been reported in 2010 because of constant use of these drugs happens vigorous approval was only the abilities. Even after the approval, after the post-surveillance market, after 10 years of the drug was again was found to have some side effects like increasing body fluids, increasing fatigue, and anemic conditions. This was reported in 2012. We are happy using the drug. In 2016, it was banned. Okay, what went on? So that's how that we have to very critically, very carefully, constantly use a drug. Our body cells have the dynamism to have the bifurcate. Even after putting somebody in the back, it still has the route to discuss, isn't it? So that's how the cells become dynamic. It will know how to, it will change over, it will shift its mechanism. Next. I told you this is the 5 essential steps for drug development, discovery and development. The preclinical research is what I told you. My limitations are with preclinical research. The university professor, the funding which I have, which I generate from the funding agency, it was around 4, which are able to only do up to the form. Now just imagine how much money is involved for clinical trials. Okay, so it doesn't mean that you should be bogged down about all my stories you have or they use. But this basic research, this is the first lead or the ladder for the drug discovery. Okay. Next. So now you could see how many hours is involved to discover a single drug. You can see how many experimentations. This is not, it's from top center for drug study of drug development. They say this much hours, enormous amounts of hours and experimentation is involved and researchers to discover a single drug. Not surprising. So many, all the people all over the globe have worked to find out the vaccine, right? Isn't it? It's not surprising. So you can see the estimated cost around 2.6 billion which will gain the market for researching and discovery. And more importantly, almost 10 times more on post-marketing business. To see whether the drugs have any side effects, to study the population effects, diversity. Another 10 times more money than yes. This is, I told you, this is what is very helpful to us. What are talking about the conventional drug discovery process. And this is how high throughput technology is practiced. This is the very latest and modern methodology for even for COVID vaccine. It's the high throughput thing. Automation robotics. That means earlier we manually do experimentation by getting out the sample. Now we have a robotic machine. Because when you do manually, you need to be highly in a sterile environment. Even your sweat, even your sweat what in your bath, you do a everything to get contaminated. Because your sweat again contain RNA, it's going to degrade your drug. We have a very clean environment or clean atmosphere to do the research, drug research. So we need a high skill. The best thing is don't use men, use a robotic. Now researchers are no longer being toiled around the lab to do research. Switch on and supervise the robots. The robots will do. Miniaturization is again is the very important. The smaller is more beautiful, right? Yeah. Smaller is good. Smaller is more effective. Hence, earlier we used to do 96 well plate. We used to look not aware about the instruments we have done. ELISA plating, to ELISA plating, there is a 94 well. If you do PCR, there is a 94 well, right? But imagine it's done again for 94 wells. That means 94 samples you can test. Okay, imagine you grow bigger, 2,500 to 2,000. That means even a single shot we can screen 3,000, 5,000 forms. That is again the beauty of miniaturization. And another thing is, all the reagents used in the panel very, very expensive. Even to a PCR, the tag polymers we use inside, 1 gram costs around 60,000 rupees. Very expensive. So instead of having the sample, instead of adding 20 microliters, if you add 2 microliters, 10 times reduction sample, the cost of experimentation, the cost of reduction is lesser, right? Again, it's money, right? So miniaturization plays an important role during the kind of. So it is time consuming, you reduce the time, screen the compound. Another one is you have lesser assay volume. That will again save you money. Artificial intelligence. This is again, the modern, even last month when I was in the studies for my ticket program. Our vice chancellor most talk about machine learning, artificial intelligence, and IoT, Internet of Things. It cannot go beyond the quality research. Our dependence on internet is indispensable. Artificial intelligence again is indispensable. You can speak from your phone, isn't it? Your voice is better. You show a hand, the volume of the phone gets lesser when you go near it. So artificial intelligence again has a tremendous impact. Even the car is automatically the wiper goes on when the rain falls on your car, right? The light intensity increases in the car. You don't have to switch on your light. So these kind of things. Example, what is that?
Alexa.
What is that phone? Siri in iPhone.
Alexa.
Alexa. Right. And again, it's an artificial intelligence. Next. So this is some why these are being chosen is that provide better data quality, process consistency and is automated. Human errors can be minimized. Big plus is that human errors can be minimized. And also provide lot of freedom to scientists to pursue. So while I'm doing research, we have a point of parameter. Especially to the Western Baltic, when I confirm control, I am working on physics. Confirm whether my plant is integrated by DNA or not. I have extracted DNA, not southern Baltic, then what should be divided. Run overnight, speak to another town. The mission should be on. Power cut, trade on, technology power cut. Those are the noise. So much of constraints on research time and we have going around. But now, a researcher can be a supervisor. See the robots are doing that. Next. Next. This is what talking about the mineralization, like increasing the density beyond 96 states of format. Simple thing. See, there's a small story I tell you when I was working with Accredited. The simple my previous quarter, my Germany counterpart told me. They're asking why the sales of the Colgate, paste was decreased. There are so many anecdotes about why it is decreased. So many questions. So the whether you have the competitor, the other competitor in the market, and they think that the thing is the essence of the stock is that don't underestimate anybody. Everybody has a point of view, and they say the person who serves speed, speak of himself during the break. This person was observing the talk, and he told that, sir, I have one suggestion. What is that? I tell you, widen up the holes in the opening mouth size of your paste. That may increase the sales. Stupid. But the person German and the German is very good at technical aspects. He is smaller than that. You see that the order that most of it. I was surprised. The sales was very high. Simple logic. Widen up the mouth. Increase the sales. The more pace you're going to. That's what the thing is. So mineralization is simple. Simply you can say that more number of samples, less number of solvents. So decrease the cost and increase the speed. That's it. I don't want to go more technical into it. Next. Microfluidic chip technology. This is again an alternative take on using the animal system for in vitro testing. Interestingly, we have the biochip from India. So these days now, like instead of exchanging horoscope, when you find a match of your life. Yeah. So you will see only the external beauty around him, right? Beauty or the other societal status or economical status. Yeah. No, so apart from that, whether you are compatible is not compatible is again the genetic compatibility. So whether the person that has been proposed has any genetic disorder, it is not appeared in this generation. It may be in this future generation. So we have a biochip to really identify, screen the entire genome. Even to the project in 2000. This is how you can sequence the entire genome. So the entire genome is kept in a slide. Put the system and then find out what all the things that you have. Put on the match. Synchronize, check as you check your eyes. You check it and see the compatibility match. No wonder it's going to happen in the near future. Okay, next. The machine learning artificial intelligence is that what we talked about the from robotics to image analysis and logistics. So the data that you are having right now, we have enormous data. Sense. We have a big data analysis. Don't think that you are right. You have to learn also computer science. As you are now aware about using internet, right? So big data analysis is a very big opening for you when you go for a job. They don't use, they won't select you only for a single domain, mind you. They will select you based on your multi-task person. Not that the jobs are not there, they are not equally talented to take up the job. That is the problem. In the institute, they talk about placement. Placement is very less deep, wide. Not that enough companies are there, they're not recruiting. We don't have enough trained manpower. So we have to put our effort. We have to diversify. We be multitask person. If you are a multitask person, you will get a job. So here we will talk about the image analysis logistics. The image quality and we can be able to even your biometric, right? Mobile phone, your face recognition, it's all on artificial intelligence. Okay, next. I'm not going into face, I will confuse you. Okay, next. So this is how I started. Maybe with 15 minutes I'll buy that. சார் நிறைய சொன்னாரு சார் என்னதான் பண்ணாரு அப்புறம் என்ன பண்ணிருக்காரு 20 years of research மேடம் சொன்னாங்க என்ன பண்ணிருக்காரு இல்லையா கொஸ்டின் வருமா இல்லையா So another area of interest I told you it's on hydrotrax. Especially my target is my target is towards identifying a lead component for diabetes. There should be some point, right? Okay. Next. So research focus is my research focus. My team research focus is on first graduate plant tissue culture and use of genetic transformation using producing rhizal genes. Induce the plant to produce more roots. In some plant, roots are important components of the drug. Where the root has the component of like we have more. The roots has the potential of the steroid, which are the potential therapeutic component. We can use that. We can express the plant to produce some therapeutic compounds to express the drug. And green synthesis of nanoparticles. So, there is nanoparticles for surprise. People talk about nanoparticles. But you see there no. They won't get research no. And the level வரல. அதான் உண்மை. Nanoparticles. அப்போ we have made method. Then the nanoparticles. We have synthesized the nanoparticles. And then we oppose under the effect. That's it. Scientific evaluation of the traditional facts. Traditional number of over and facts are expected. But the time again it gets distinguished. People are not known for the effective therapeutic application of this facts. So your point was that network pharmacology for commercial drug formulation from the Ayurveda. I think when you give the compound is able to control diabetes. 30 years. But people never know what is there inside the compound and how does it cure diabetes. Not people suppress diabetes. In the company people never know. As long as the formulation or sonam says, they have to, right? So we are into more on scientifically validate. That was one of my piece. So all this focus are one of my biggest work. Okay. So the challenge that we face in traditional medicine because of lack of standardized regulation process. We don't have a standardized regulation process. The moment there's no standardized practice. There's no records, clinical clinical data that prove how it proves and what is this success ratio. Nobody knows. Then a toxicology, no toxicology test. Just here, if we have any side effect, we can raise toxicology. There's no toxicology testing. Lesser side effect, scientific validation of that is an issue and challenge. I took up the challenge. My team is working on that now. Next. And I told you this is where my protocol works. I work on Android probe. It's very anti-diabetic. These are they call anti-diabetic plant. Plant based Asako. Common name. I'll tell you. So this plant has been used for anti-diabetic time module. But they don't know what is the active components involved. My interesting nature of how to explore what is there in that plant. Then Petrosia, this is the main. We have Petrosia, Victoria, another plant which is found in Kolimale, exclusive of Kolimale. We have Petrosia, panic meta has effect. Our is proven. This plant does not have, so we are trying to do that. Explore what is there. And one more student will join me for the PhD from Orissa. That's all how she located me. As she located me, I don't know how she located me. And then she came to our plant club years around from the Guru Ganga National College in Orissa. Took the plan and came down the road to do research on that. This is on marketing. And of course, I told you one more. My latest PhD students work on the mechanism, the polyhedral formulation. I told you, it was able to cure. It was curing disease for the past 20 years. But still, the scientific validation was not there. And God, her research, her 5 years work has given some insight, about what is there in mechanism, which was able to control that. And my later research projects on ginger have been prominently used even for the COVID. My focus was not on COVID, my focus was on diabetes. I found that the compound ginger has an ameliorating effect on controlling diabetes. So this is what is my area of research, what I've been doing in the past 2 decades. Next. So what I told you, it's my thing. I'm showing in picture what I really did with my students. So this is the plant focus for Perianage, okay? Perianage and Perianage. So this plant is from been used for ancient folk medicine, then is identified in politics, and we have screened, isolate, and characterized active principle, and get some molecular document studies to identify what is the compound and how compound the compound over there. Next. So I told the in vitro using the cell line studies, I found that my extract of my plant was found to be effective. I just go through the slides. Next. This is I told in vivo using the animal model. When I use the animal mice model, I again find that the 200 mg per kg of ETL means ethyl acetate, ethyl alcohol extract of Andropophis lineata. Okay. When I take the plant extract, when I tested with the rat, the rat was found to induce diabetic on the rat and then treated the rat with my compound. And found it was effective. Next. And the very effective, taken the pancreas. Now pancreas is one which will produce the insulin. Okay. So I have done the experimental studies and find out that I treated with my extract of showing. The restoration of the isolates of pancreas in the pancreas was restored. So this is one confirmatory study that my cousin got.
Yes.
Compound alone. Yes. So when I did some analytical study to structure, and it again structural elucidation. What is the compound? When I do a biochemical tool to structurally elucidate the compound. If I find dimethyl 3,4 tetrahydroxy, succinate is the compound that I have isolated. This is the compound which I thought responsible for the anti-diabetic effect. You got my point? From the crude extract, from millions of compounds, I told you, right? I narrowed down to a single compound which was able to give the diabetic. Then I told you I used PPR gamma, the diabetic targets. And this is on in silico analysis. That means on using biotechnology schools. I told you I use rosiglitazone, the standard drug, when I try to dock. using a molecular software system, a software. So I cannot do a virtual, manual screening of all the compounds is very expensive. So I do the in silico approach using bioinformatics tools. And I found this compound was exactly binding to the receptor which I intended. And therefore, I perceive that it will be effective when you go for a clinical test. Got everyone? Yes. This is again the how I visualize the 3D structure using the bioinformatics software. Next. Again. So I told you this is what the summary of the finding is that the crude was find very good antibiotic effect, and the docking score we used can identify the purified compound. It was as similar as the standard drug clozaprazone, and this how this may be a way for the discovery of the drug for diabetes. This is substantial. Whatever because without publishing in a journal, it's useless. How good research area that reflects in the publication. So my student, the first student, Deepa, she is published now. She is Russian professor in NAAC, Andhra Pradesh. So she has a further work and she published a paper. Next. And this compound, what she isolated, and then she was able to suppress the hypothesis. One more diabetic target, she published in the journal. Next. The next is I told you one of my students again, he worked on using the Victoria plant. This again to the proteins of nanotechnology. Next. And he approached in a different aspect. He used silver nanoparticles. The first student would extract and purification. Next student will more improvise that using silver nanoparticles. And we found that was more effective compared to the food extract. So if we put with the silver nanoparticles, the activity was more. What we thought? Next. And since that plant is an endangered medicine plant, they have to micropropagate its plant. That's what we did. And again, we had an interest that why you going for cancer, we tried again for the cancer anti-cancer activity. It's an in silico analysis. We found that it has using the Molbro, virtual Molbro software. We found it has also a compound which can be used as anti-cancer drugs. It has anti-cancer property. Next. On the first report, we got the quercetin. Identified a compound called quercetin in that and found that it may have a potential target for cancer target. And this we have been published. I told you this green synthesis of silver nanoparticles. That is one great student thought that published materials that as well as international publication. It's well received. We have more than 102 citation in that. And he found that regulating role of ethyl acetate, that is from the food extract, and how he control the carbohydrate. And he published in the biomedicine and phytotherapy. Okay. I told you my research student worked on the anti-arthritic, and he found that she has collected 2 species from Eastern Ghats in Orissa. Rheumatic arthritis. Next. Of course, you did the plant tissue culture, and then we have improvised that. We tried to now we tested gold Next. So this is how she has micropropagated the plant, and then use green synthesis using the gold nanoparticles. Okay. And we found that was more effective, far more effective compared to silver. This is how we have to improvise. Okay, and she has identified purified compound of uric acid and we are still yet to publish that paper. But anyway, it was well received by the research community that the gold nanoparticles was far more effective and more lesser toxicity level compared to silver. Next. That is a synthesis process of the gold nanoparticles. Okay, next. This I told you the scientific validation of the polyhedral formulation mechanic, which they have been used almost 2 decades as a drug formulation by Siddha, Ayurvedic practices. and it was being given to the patient. But they never know what is there in that composition. So we underwent the molecular mechanism of all the compounds present in that mechanism, which was responsible for the controlling diabetes. So we did various extraction process from soft slate to microwave extraction process to extract the metal. So the challenge here is that earlier we talked about single plant. Here the formulation is 5 different plants. 5 different plant extracts, it's a little more complicated. So we went into network pharmacology approach because we cannot manually screen such a big volume of data. So what we did is, like, within the software, we have network pharmacology. Each plant we have identified these PCFs, screen, we identify the compounds, then pull down. We got my point. So it's a hell of a lot of work. It's all the HPA analysis of the mechanism and then identify all the important compounds. So responsible for that. And then more importantly, you didn't went for gene level expression. We thought study what are the diabetic pathways, how what are the genes, how the genes are responsible to controlling diabetes or they are affecting diabetes. We thought that one gene is called the carrier protein called glucose, which is responsible for transport of glucose to the cells. And if glucose is not transported, the glucose remains in the state that again increases your blood sugar. What we want, glucose has to be transported. So for transport, we use the glucose expression. And we were able to find out an expression level. We got a good expression level in the mechanism. We found that glucose expression was genes upregulated and it was more effective. And we did, I told you, find a pharmacology, the approach on using the wall grow software and identifying with the narragin with the compound. Found as an excellent bonding and there's an anti-inflammatory effect what again achieved in the next step. And diabetic and anti-inflammatory. And this was well received when you published, of course, a lot of struggle to publish in National Product Research. And 2019, and then she published it. This was well researched for impact factor. Next. And there's another DPT project where we work on, I told you, ginger, that we look at gingerol in the other different perspective. Not on virus, antiviral property, we went for antidiabetic property. And we found that gingerol was again effective in as an antidiabetic agent also. So we tried with different ginger. We found the Tamil ginger was more effective than the Odisha cultivar. And the gene expression again, the Tamil variety was more good for gene expression. You identify the active compound using DC. More importantly, you identify important compound called D. Next. So this we published in Book Biochemistry Journal. It's well received. Of course, we didn't restrict ourselves with anti-diabetic. We went for anti-diabetic also. We found that one of my students worked on it. Really, he published a very good paper on that. Impact factor of more than 5.6. This is the paper on food and function where we use. This is a work of an textual. This paper was a work of an textual source. Now he is in IIT. It's not that very important. What I'm trying to tell is that no matter where you are, what you are, if your research quality is good, you will be treated according to your method of your research. Now he is an IIT Hyderabad. And this is a paper he published. It is good for I told you, good for activity using this is again in the next level of biochemistry, which again has been one of my collaborators called Krishna, he's an IIT professor, my collaborator, and Subhadi is from SOA University in Pune. Research is one ground, not that you next door your friend is your, you may be a mate, but cannot be a collaborator. Research interest may be varied. What may you want? You may be a good friend, but cannot be a collaborator. Collaborator is one. I got collaborators all over my country. See, one I shared with them the PhD. I was examiner for a student. tractor and we got to know, and then he supplied me within the sense of it's not only my work, my student work. Now you imagine what I told you. The thing. Next. And this is again that's been the most highest impact factor I got is around 7 in journals where we talked about the chemotherapy of the student work. And this is very surprisingly done. This my first PhD student. You see, after almost 10 years of this research in 2021, we published a long group of paper in critical reviews of science. This is impact factor around 1, so it's highest impact factor I will say. Based on my ex-student good research, after 12 years of research, he has published. So the weight is worth, right? So what I want to say that is our research perspective is the increase in the demand of hormonal properties in oil for the world because of reduced side effects, because allopathic drugs having a lot of side effects, using the medicinal plant or the fiber plants reduce the side effects, and authentication and scientific validations, and economical approach of the fiber drugs is very important aspects in this present scenario of drug research. Okay, thank you. Is there anything or any questions that you ask me?
Actually, doctor, allopathic drugs suppress diabetes எந்த இந்த மாதிரி நம்ம பிளான்ஸ் பண்ணி எடுக்கிறதுல அதுவும் சப்ரஸ் பண்ணா ரெக்கவர்
Use the word controlling. Diabetes can be only controlled, it cannot be cured.
So any other disease, I mean, allopathic எடுக்கிறதுக்கு அந்த cure பண்ணாம சப்ரஸ் பண்ண நிறைய சப்ரஸ் பண்ணாங்க சப்ரஸ் பண்ணி கண்ட்ரோல் இந்த மாதிரி நம்ம ஃபைட்டோ அந்த மாதிரி பிளான்ஸ் போகும்போது is there any way to cure the disease? Or these are also
allopathic drugs is cured. You cannot undermine that. See, these allopathic drugs are otherwise called magic bullets. Magic bullets. It hits a particular target. The Nature report 2000. It's called magic bullet. You hold a magic with a particular target. You want to cure diabetes, you have a particular reward and receptor. The hit is perfect. Because of the hit, it shows. But the problem is here, if you're going to continuously hit on the target and again, it takes a bite, the cells are dynamic, it points to bifurcate. That is the issue. But when you talk in terms of the allopathic drugs, with your conventional ayurvedic and traditional medicine drugs, this will have a continuous process which will have relatively take more time when compared to allopathic drugs. Why it takes more time? Because this 50 target only has a specific target, whereas the herbal drugs, it will have multi-targeted approach. Here I talk about a magic bullet, single target. Here multiple targets. And because of the multiple target, it goes and hits with the other targets also. Thereby, reducing the side effects. I put the word reducing the side effects is not more side effects.
So targeting multiple targets.
When we talk about the immediate action, acute inflammation or acute disease, there is no other go for allopathy. When you go for a chronic treatment, when it is in the middle stage or in the primitive stage, you can go for advanced treatment. But when it is in acute, we need to hit the maximum.
Thank you, sir.
days, but biochip is used to identify the disorder. What about microchips are associated? Both are same or
Microchip is different for the drug discovery. When you really go to microchip, we have a pool of compounds. Okay, the compound dynamics is being integrated in a chip, and when you go for in silico screening, you can identify the target whether it's a particular target or not. Like in silico analysis, the next stage is silico. Then after the microchip, then you can go for your preclinical trials. This is another step to narrow down the research. Biochip is one, you can enter the sequence of genome, and then you can cross-check with your sequence, the disease sequence, or inherited disease. Using reducing obesity is a big business. It's a very big business. Obesity related to adipose tissue population. The one result we found that reduce the adipocytes. Not with microbial cut. I'm not sure about that. But anyway, but adipocyte differentiation. If you can limit or cut short the adipocyte differentiation, then you can have reduced the obesity. Obesity mainly, apart from the theoretical level, is most on your physiological condition and environment aspects. So one common question was asked to me using the broiler chicken. There was one Suna chicken people who I was releasing on the phone. Intuitively, I asked him, is that you are checking any growth hormone so that the chick is growing faster than 45 days. Sir, how is it possible, sir? 1,500 bucks, sir. 1 kilo chicken is only 200. 1,500 rupees, sir. This hormone injection that I take, then how will be economically feasible? He said, I can give you only an antibiotic shots that prevent other infection, not on growth. But how it's growing? The logic is very simple, sir. Give them food, give them no place to roam around, restrict the space, they will become big. Same here. Input is there is no output. There is no physical activity, right? The chicken put in a restricted area, give them food. Restrict is movement. It becomes big. That's how the body chicken gets big. Not because of injection. So, control is so see, even whatever the drug, if you don't move, if you don't do physical activity, hit the gym or whatever the exercise that you do, it will be tough, right? But there are some movies also came, reduce the effect. They have also liposuction. Now we have because people don't want to work hard, they don't want to go to the gym, don't want to do any physical activity, so they want to reduce the effect, reduce weight. Now we have the liposuction. They will support the lipids. Some negative effects also. You need to have a trained hand for that. There are people who die. So even hand transplant, they might. Certain things what we if you are with nature, go to all of it.
Sir, you said that biochip can be implemented in future. Like it can use to detect the result. Yes, sir. Like there are so many disadvantages also that can be inserted in our body and it.
Not that what you see.
Now some researchers are going to like biochip.
Biochip to detect as we have GPR detection. That is one thing your mobility. That chip is different. This is the chip talking about the entire genome, not your genome, the genes. We have around 30,000 genes. So these genes are sequenced We already sequenced in 2000 itself, 20 years back itself, we have sequenced. We know MPS genome sequence. That sequence can be miniaturized, I told you that, miniaturized in the form of a biochip. You know MPS genome sequence, we know what is the disease sequence. And then we can really find a match out. And that is the reason even now after the discovery of the human genome project, after the identification of MPS genome, it's an astonishing fact that really we consider humans as the most advanced creatures on the earth. I showed you a small bomb last year against various slides. No, that has 1.5 times more gene than us. Wow. Onion has 110 times more gene than us. Not that more genes are more advanced. Even among the 30,000 genes that we have linked in genes, 90% are junk. Useless. Functions are not known. Only when we have 10%, we have 50. And more importantly, you know the Y chromosome? The manly Y chromosome. I'm sorry, my friends, Y chromosome doesn't have anything. No property which is genes which are really functionally active. That is the reason that people say female of this species is most dangerous and vulnerable and most important in biology. Male presence is insignificant. I use the word insignificant. And if you just analyze what is there in Y chromosomes, they have only 50 genes. And 50 genes, there is only certain genes which are like hypertrichosis, called and emergency of mental. That's true.
So, women are more useful.
Exactly. Oh, yeah. Oh, sir, college. When we get into I'm sorry, Mother. No more questions. I have to understand.
So, what is the which helps in reducing that?
What is, you know the famous statement from Tiruchirappalli? Terima, guys. Tiruchirappalli guide பத்தி தெரியுமா? உங்களுக்கு யாரும் தெரியாதா? அவரோட ஃபிட்னஸ் காரணம் என்ன தெரியுமா? ஃபிட்னஸ் காரணம் தெரியாதா? ஜிம்முக்கு போறாரு அதான் ரைட். Whichever is palatable to the tongue. Don't take food for your tongue. Take food for your body. எதெல்லாம் bitter-ஓ, that's not good for your health. But too much of taking that thing again happen, and then take it. The limit for everything.
Thank you, sir.
They are also very restless now. Okay, thank you. Thank you. I'm very happy that. I'm happy for two reasons. One is that you're still bright enough to listen to me. I'm more happy because in spite of the evening hours, in fact, the time minutes, in spite of your friends' annoyance, you're asking me questions, right? Happy. Something like talking. Thank you.
Thank you, sir. We really had a productive and inspiring time together, and this event is about to come to the very end. Hope you have found the presentation on this conference very informative and helpful. Let us all be guided by all the things we have learned and heard through the conference, and we are able to see the influence in our future. It's time for tea break. Have your refreshments out in the hall. Please come back at 5:05.
Kindly requesting the gathering to please assemble back to the hall at 5.
Please assemble back for validation.
Online participants, please be back at 5 p.m. and it's tea break now. Online participants, please be back at the 5 p.m. and the validation program will begin. doesn't set limits to imagination. Creativity is thinking of new things. It is doing new things. Innovation is the ability to see change as an opportunity, not a threat. Assalamu alaikum wa rahmatullahi wa barakatuhu. And a very good evening to one and all gathered here. Here, we are in the final phase of our national conference, PCBA 22. So let's begin the valedictory event. Prayer should be the key of the start and love of the end. I call upon Aquila of second UG Biotechnology to recite a few verses from the Holy Quran. Assalamu alaikum wa rahmatullahi wa barakatuh. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين. الرحمن الرحيم. مالك يوم الدين. إياك نعبد وإياك نستعين. اهدنا الصراط المستقيم. صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين. آمين. صدق الله العليم. السلام عليكم ورحمة الله وبركاته.
Thank you for praising the occasion. Welcoming newness, welcoming charm, welcoming integrity, welcoming venture. More than anything, welcoming people, because at the end of the day, all that matters are the souls that you have earned. I kindly request member in charge of the department, Dr. J. Sebastian Raj, sir, to welcome the gathering. Please, sir.
All praise to be Almighty Lord. The fresh beginning always exists. We only have to welcome them. Every experience in your life is being orchestrated to teach you something you need to know to move forward. A pleasant evening, one and all present here. Respected dignitaries on the dais of the dais, I deem it as a great privilege to welcome you to the valedictory function of National Conference on Post-COVID Complication, a Biotechnological Approach. Today, a pleasant evening. It is my great pleasure and honor to welcome our Honorable Vice Principal, Dr. A. Mohamed Ibrahim, sir. I welcome you, sir. I feel immense happy to welcome our chief guest, Dr. P. Suresh Kumar, Professor and Head, Department of Biotechnology, Anna University, Trichy. I welcome you, sir. I feel grateful to welcome our Dr. Samsudin, sir, MID of Zoology Department, and also I welcome Aslam, sir, from Botany Department. It is my honor to welcome Dr. Padmanabhan, Research Advisor, Jamal College in this occasion. He participates from Germany through online mode. I welcome you, sir. I welcome our coordinator, Dr. A. Sadiq Buhari. I welcome you, sir. I am very much happy to welcome 8 staff members, research scholars, and students of microbiology, botany, zoology, and other sister departments, and also staff members from various colleges in this occasion. I welcome you, sir. I'm happy to welcome Dr. Anisa from Zoology Department, Jamal College. I welcome you, sir. I feel great privilege to welcome the active participants through online and offline from various colleges and university faculties, students, research scholars in and around India to our national seminar. I welcome you all. I congratulate you. People are the real reason for success of this program. We feel great privilege to welcome the great pillars of our department, Dr. T. Nadu Begum, Dr. Gopala, Dr. Deborah, Dr. Nadu, and Dr. Andrea, my department professor, without whose support such programs would not be possible. I welcome you all. I welcome Mrs. Mafia ma'am from Zoology department for this occasion. I welcome you, ma'am. Stop dreaming and get moving. People of accomplishment rarely sit back and wait for things to be acted. They went out with determination to make things happen. Hard working is the price of success. You can accomplish anything if you pay this price. I congratulate and welcome our organizing secretary, Dr. S. Benazir Meher. I welcome all of you once again into this pleasant evening of this national seminar. Thank you all.
Thank you, sir, for your warm welcome. Feedback promotes personal and professional growth. Feedback is about listening actively, taking the time to analyze, and then thinking of the best possible solution to perform better. And this is the time to share the experience of past 2 days of a national conference on PCBA 22. Here we have Anita ma'am from Srimati Indra College.
I thank the Almighty for this gracious occasion. I thank Organizing Secretary Dr. A. Sadiq Buharisa, Convener Dr. S. Benazir Begum ma'am, and need of our topic on post-COVID-19 complication of biotechnological approach. The organizing secretary invited great scientist Dr. B. Ramalingam, ICMR, Dr. Nadu, CSIR, Dr. John from RGCB, and Dr. Kumar, IIS, Dr. Jayakandan, Alakappa University, Dr. Today's chief guest, Dr. P. Tiruchirappalli, Anna University. Treatment in India is based on protocols set by ICMR scientists. Treatment இப்படிதான் கொடுக்கணும் டாக்டருக்கு செக் பண்ணி கொடுத்தது ICMR scientist. Okay, first waveல throatல இருந்து respiratory systemக்கு லங்ஸ்க்கு போக கூடாது அப்படிங்கற ஒரு கான்செப்ட். அப்புறம் இந்தியால இருந்து ஆல் ஓவர் தி வேர்ல்ட் ட்ரக் போக காரணமா இருந்தது சயின்டிஸ்ட், நம்ம சயின்டிஸ்ட். இன்னொன்னு ஆன்டி வைரல் ட்ரக் ட்ரீட்மென்ட்க்கு கொடுக்கணும் அப்படின்னு ஒரு இத கான்செப்ட் வச்சது ICMR சயின்டிஸ்ட். செகண்ட் வேவ்ல ஆக்ஸிஜன் டிமாண்ட் இருக்கு, அதனால உயிர்கள் இழக்கப்படுதுன்னு சொன்னது நம்ம ICMR சயின்டிஸ்ட். So now the post COVID alert and awareness கொடுத்த நம்ம biotechnology department and the principal, vice principal, Jamal Mohamed College, the thanks சொல்லலாம்னு நினைக்கிறேன். If you accept this concept, of biological science, convener, committee members, and also the college principal and vice principal of the college. Thank you, sir.
Thank you, ma'am. It's time for feedback from students.
Good evening to everyone. The conference was very innovative and very Good evening to everyone. The conference was very innovative, and I gained a lot of experience and information from this conference. I thank the team and everyone who was involved in organizing this wonderful conference and giving me an experience and a wonderful opportunity to attend it. I thank you all. Thank you. I'm Lavanya from BIOTECHNOLOGICAL College of Arts and Science. I'm Lavanya from BIOTECHNOLOGICAL College of Arts and Science. I'm really thankful to your management for creating this seminar. It's very useful and informative to us. We know more information about COVID-19 and lot of ideas about students. And really thankful for your management. Thank you.
Good evening, everyone. I am Mr. Jamal from Bharat College from Tanjavur. I am glad to thank JMC College to give this organization a very interesting and excellent national conference. Thank you for the excellent conference possibility to attend. I really enjoyed my time at this college to gain more knowledge about the post-COVID-19. Thank you to everyone.
Thank you all. Thank you for your valuable feedback. Report presentation is continual dealing and continual calculation of development. I kindly request the organizing secretary, Dr. S. Benazir Begum ma'am, to present the report.
Assalamualaikum. Good evening to one and all gathered here. It's my privilege to propose the report for 2 days national conference on post-COVID complications, a biotechnological approach, PCBA 22, which is sponsored by Department of Biotechnology, Government of India. The session began with a welcome address delivered by Dr. A. Sadiq Buhari sir, convener, Department of Biotechnology. The Honorable Principal, Dr. S. Ismail Mohamed sir, delivered the presidential address. Our esteemed Secretary and Correspondent, Sir Haji Dr. A.K. Kaza Nazimuddin, and Assistant Secretary, Dr. K. Abdul Samad sir, delivered the felicitations. The release of the seminar proceedings was done by our Secretary and Correspondent, and the first copy was received our Chief Guest, Dr. A. Ramalingam sir, Scientist E, Department of Immunology, ICMR, Chennai. The other dignitary members also joined in. The Honorable Chief Guest, Dr. A. Ramalingam sir, delivered the inaugural address. The vote of thanks was proposed by myself, Dr. S. Benazir Begum, Organizing Secretary, Assistant Professor, Department of Biotechnology. So the first session began with the plenary lecture 1, delivered by Dr. A. Ramalingam on the topic COVID-19 sequel, visit major concern. It was followed by a plenary lecture 2 on targeting the pandemic of the century, insight into novel approaches by Dr. John Bernard Johnson, Scientist E1, Department of Pathogen Biology, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, Kerala, through virtual mode. After lunch, plenary lecture 3 was delivered by Dr. Kumar Somasundaram sir, Professor and Chair, Department of Microbiology and Cell Biology, Indian Institute of Science, Bangalore. Topic: Next Generation DNA Sequencing, Implications in COVID-19 Pandemic, through virtual mode. It was followed by the paper presentation from interested participants of various esteemed institutions through blended mode, which was happened with the judges, Dr. Salahuddin sir, Department of Zoology, Dr. Aslam sir, Department of Botany, and Dr. T. Nargis Begum sir, Department of Biotechnology from our college. Nearly 70 participants presented their research papers from various institutions all over India. In the second day, began with plenary lecture 4 on potential of bacteriophage to fight against antimicrobial resistance to combat COVID-19 by Dr. Nadesan Monikam sir, Senior Principal Scientist, Environmental Biotechnology Section, Industrial Toxicology, Research Center, CSIR, Lucknow. It was followed by the plenary lecture 5 on explore the novel mechanism for inhibition of cell wall biosynthesis, which was given by Dr. J. Jayakandan sir, Professor and Head, Department of Bioinformatics, Alagappa University, Korekudi, through virtual mode. It was followed by models. Models were displayed in Zoology Seminar Hall from interested participants, which happened in front of the judge, Dr. I. Joseph Anthony Gerald, sir, Head, Department of Zoology. It was followed by the poster presentation from interested participants of various esteemed institutions in blended mode in front of judges, Dr. T. Nargis Begum, Ms. H.F. Syed Mafia Hanif, and Aslam sir. It was followed by the last plenary lecture 6 on biotechnological approach in drug discovery and development, given by Dr. P. Suresh Kumar sir, Professor and Head, Department of Biotechnology, Anna University, Tiruchirappalli. Now the valediction ceremony began with the welcome note delivered by Dr. J. Sebastian Raj sir, Assistant Professor and Member in Charge, Department of Biotechnology. Our Principal, Vice Principal, Dr. A. Mohamed Ibrahim sir, going to deliver the felicitation. The valedictory address was given by Dr. P. Suresh Kumar sir. The report of the seminar is presented by myself, Dr. S. Benazir Begum, Organizing Secretary, on the topic Post-COVID Complications, a Biotechnological Approach, PCBA 20B. Then the vote of thanks, the distribution of certificates will be presided over by our Vice Principal sir and our Chief Guest. The conference witnessed the overwhelming participation of nearly 300 participants or participated from all over the India. The vote of thanks will be proposed by Dr. T. Nargis Begum, Department of Biotechnology. So the seminar will conclude with the national anthem. Thank you all.
Thank you, ma'am. To light the lamp of spiritual wisdom in our hearts, you need the container of attachment of 1 point concentration, matchstick of wisdom, and the oil of devotion. All in one, we have a dynamic Vice Principal, sir, A. Mohamed Ibrahim, sir. We welcome you to address the gathering. Please, sir.
Great indeed is the grace of the Almighty Allah. Alhamdulillah. Most respected Dr. P. Suresh Kumar sir, Professor and Head, Department of Biotechnology, Anna University, Tiruchirappalli. Distinguished Dr. Sadiq Bukhari sir, Dr. T. Nargis ma'am, Dr. Sebastian sir, Professor Benazir ma'am, my good friend, Dr. Shamsuddin, Dr. Aslam, other potential staff members of this great department, staff participants, student participants from all over India, So all, I used to say, the jewels of Jamal. Assalamu alaikum. A very pleasant evening to all of you gathered here. It gives me great pleasure in joining this department, which has offered a wonderful intellectual feast for a couple of days. PCBA has become an ECBA. We always say Jamal means beauty. Jamal means beauty. The Department of Biotechnology, BT. There is beauty in BT. Okay. That is the reality. Because you're all great personalities. So we always pat on our back. We deserve it, I say. Jamal is situated in the toll gate, but we produce Bill Gates. Number 7, Race Course Road. That is the college postal address. All of you may know very well. Number 7. Jamal is not simply 7. Jamal is a heaven for heritage. Jamal is an educational treasury of Trichy. Jamal is the Taj Mahal of Trichy. Jamal is an academic paradise. Jamal is an intellectual powerhouse. See, when I refer to all these things from college, because Jamal doesn't become an intellectual powerhouse all of a sudden. On the highway of higher education, we have crossed several milestones in the journey of 70 years. This is the 75th year of our academic journey. In the past 75 years, we have many scientists, many stalwarts, many superheroes shining very well in the academic field. So that is why we have reached the status of excellence. What is the potential for excellence? So that way, the Department of Biotechnology has to be appreciated and congratulated. The organizing secretaries, the organizing team, for example, Dr. Sadiq Bukhari. Our Sadiq Bukhari illa, Sadiqum Bukhari. I thank the teachers and students who have given a wonderful feedback. The feedback is the proof of the success of this conference. I appreciate you all for the valuable and wonderful feedback. The secret of the success of this seminar, conference, organizing a conference that of this size, in blended mode, also in visual and virtual mode. In real, in flesh and blood, you are presenting the papers, person presentations, variety of academic activities that has attracted the attention of all the participants. Okay, the teamwork of the entire department. That's why we say TEAM stands for the expansion goes like this. Together, each achieves more. TEAM expansion is together, each achieves more. So every staff member and every student of this department, they come together, join together, they thought together and worked out together. Togetherness is the secret of success. So together they have achieved a lot. They have refreshed all of you. That is why I said they have given an intellectual feast in these 2 days. Post-COVID, the word very COVID is threatening, you know. Many Davids have gone due to COVID. David, no David, no David, because of COVID. Okay, they're all terribly afraid. They're waiting for the end of this virus, you know. So they're afraid, terribly afraid. So thanks to God, thanks a lot, and they have found out a solution with all great and nearly 6-7 scientists. They have addressed the gathering. They have given a lot of inputs in these 2 days academic proceedings, and I was told nearly 250 participants all over India. So a big thanks to all the 250 participants. The staff and students, thank ma'am, especially for wonderful feedback. So without participants, nothing is possible. So the participants are the real beneficiaries, and organizing team, all the staff members, they deserve congratulations and appreciation. I congratulate the Department of Biotechnology for having organized such a wonderful national conference for the benefit of student community. And I thank the resource persons, all the resource persons deserve appreciation, especially Dr. P. Suresh Kumar sir. P. Suresh Kumar, PSK. SK Suresh Kumar SK. So Dr. SK, we makes it okay for the rapid address. That is here too. So with these viewers, it did not take much of your time. I know your mood. You are eagerly waiting to receive a certificate and make an exit from Jamal Mohamed College. So I know your mood. So it did not take much of your time. I thank all the participants. I congratulate the Department of Biotechnology. All the team members. Thank you very much. You all the best. Thank you for the opportunity.
Thank you, sir, for your valuable words of motivation. Honoring is our tradition. It's our passion and not for fashion. It's just a little we could do for all you have done. Now I kindly request our Vice Principal, Dr. A. Mohamed Ibrahim, sir, to honor our guest of honor, Dr. P. Suresh Kumar, sir. Please, sir. Short and moment. Thank you, sir. Now I kindly request our coordinator of our department, Dr. A. Sadiq Bukhari, sir, to honor our Vice Principal, Dr. A. Mohamed Ibrahim, sir. Please, sir. Thank you, sir. Every thought you produce, anything you say, any action you do, it bears your signature. Now, I kindly request Dr. P. Suresh Kumar, sir, to address the gathering. Please, sir.
Good evening once again. Okay. Pleasure to be here. I know it's my third time to be in this room. Okay. Prestigious institution. I have observed and I watched this institution right from its childhood, being a native of Trichy. I think most of the value address your vice president of the age. I wanted to speak. He has illuminated the audience. One thing I really like about the journalism of Jamal, the captain is really good. In fact, he made a small anecdote about me. SK is okay. Okay, yeah, this is the dying moment of this conference. I don't want to elaborate more. I've talked at length about almost 1 hour. So, this institution has an history. This is not something like JNC Jolly Jamal, okay? Yeah, Jamal. Yeah, what anecdote I give you? it's again, it's good to talk about your own experience to really to make you feel better, rather than something which is talked about something somebody said. I remember one person in your college who did his MSc and next year. His name is Sham. We changed his name. His name called Nadu. We named him Sham. I did my MSc in CBT. That time he was a PhD scholar. Sorry, his name is Shanmugavelu. We made him Sean. So he came from Trichy. He's from a small village down Trichy. Did his MSc zoology here, and MPhil also here. He joined for a PhD in Center for Biotechnology and NAAC. And I was doing my MTech, he was a PhD student. One Kundra Jairam, I heard about her. She is the one who really taught in biotechnology, technical aspects. Where we are now serving in an institution now because of her. And she was my teacher. She famously told, of course, Shan has a problem with his own communication because he is from the left side. He will call DNA as TNA. No wonder when everybody started to make fun of him. KJ has a huge respect for him. And one professor, I forgot his name, in the zoology department, when he came to see over him, I was there with KJ, I was the class representative for MPhil. KJ came down, KJ is a bit sort in biotechnology. He came down and greeted the professor and told him, we have never seen KJ at Accredited. She was a BBC IR official, she both stadium and fire. Such a bold lady she is. Such a dynamic lady. If you not put dynamic, you can equal to that. Even vice chancellor, she will not have a second thought of it. She came so humbly down. I forgot the name of the professor, I forgot. He was already a professor. And greeted him and said, thank you, professor, for being such a wonderful student. You got my point. Playing your zap, like we thought so much of I don't have things are there, but this now he lives in US as a NRI. Was my good friend sitting here. No, it's not Tangavel, it's Shanmugavel. Oh, Tangavel. Oh, correct. So much. I'm so sorry that you thought about that. Fact, because so good friends. And see, one thing is that he has break down the barrier of sophistication, modernity, and style, and accent, and language. These all things impress KJ with his science, his knowledge. That is more important rather than your how style you speak or how I found out you be in the environment. That is immaterial in science. Is that he is from this institution. So you are sitting in the same place. No stand from you. We modernized him by name. He modernized his research. You got my point. So that's why I tell you that article I was like, you have seen so many scientists given you delivered and deliberated lectures. What does that implicate? Not that everyone is going to do research in COVID. Even I myself, I can do research in COVID. My areas in that. I told you straight away. What does that conference brings in you? Why this conference has been outlined? What for? Then you want everybody to be a product scientist? No. To implicate a research, a technical know-how on the current modern aspects or on the trivial aspects of the society. That's the reason they taken a topic on post-COVID complications. But this is very important that you have to create an environment, create an exposure. The good scientist that you have been there, you have been witnessing for the past 2 days. Some information, some inspiration from any one of them. Your college is searching you to be a research scientist. Hope that this conference is one of the key. This has deliberated in you a certain wake-up call. to focus on yourself, not only in COVID research, in any other research of your interest, and we particularly bring glories to the institution as standard. Okay, thank you and best wishes.
Now it's time to honor the team people who have made events of the 2-day conference a grand success. The honorable judges of the poster presentation, oral presentation, and modules. Now I kindly request our coordinator, Sir Dr. A. Tiruchirappalli, to honor Dr. A. Aslam, Assistant Professor, Department of Botany, with certificate and membership. Please, sir. Thank you, sir. Now I kindly request our chief guest, Dr. K. Suresh Kumar, sir, to honor Dr. T. Nadu with certificate and membership. Thank you, sir. Now I kindly request our Vice Principal, Sir Dr. A. Mohamed Ibrahim, sir, to honor Sayyid Mafia Management with certificate and membership.
Thank you, ma'am. Thank you, sir. Thank you, ma'am. Thank you, sir. Unfortunately, the clock is ticking. The hours are going by. The past increases, the future recedes. You don't have to be great to get started, but you have to get started to be great. Now, we will announce the best oral presentation and poster presentation and the prize winners. Now I'll start with the online participants. The oral presentation, which was held online, the first prize goes to Dr. G. Chelladurai. The second prize, Ms. Semsa Damla Au. And the third prize goes to Dr. Monica Mignani, and it's also shared by Jennifer and Ms. Christie. And now the poster presentation, which was held online. The first prize goes to Soumya, slot number 12. Second prize goes to Monica Jaital, slot number 4. The third prize goes to Ghazala Shaheen, slot number 3. The certificate will be sent to you soon. Thank you all. So now let's move on to the offline presentations. The oral presentations, the first prize goes to Dr. S. Saranya and team. Rajaratnam University. For the winners, please come forward to receive the award. And the second prize goes to Mohamed Irshad, second MSc Zoology, Jamal Mohamed College. The third prize goes to Ms. Nitya and team, Bishop Heber College. Congratulations to winners for the award presentation. Next, we move on to the winners of the poster presentation. The first prize goes to Marilyn Titus and Team Bishop Heber College.
Excuse me. Cameraman.
The second prize goes to Miss Anita. Miss Anita from Team Bishop. Third prize, Panmati from National College. I congratulate all the participants. Now, here we have special prizes for the best effort and presentation. The first prize in poster presentation goes to Pugal Nila and Mamtaj Nigam of 2nd MSc Biotechnology, Jamal Mohamed College. And the second prize goes to Reem Jalaluddin and Amsa of 1st MSc Biotechnology, Jamal Mohamed College. And the third prize goes to Ms. Suhaina and Ms. Farhana of 3rd MSc Biotechnology, Jamal Mohamed College. Congratulate the winners.
Next, model presentation. First prize goes to A. Zumana and Team 2nd BSc Biotechnology, Jamal Mohamed College. Second prize goes to S. Shaheen and team for Biotech, Mohamed College. Third prize goes to Mariam Aisha and team for Biotech, Mohamed College. Second Biotech, Jamal Mohamed College.
Thank you, sirs. And I congratulate all the winners. For the offline participants, here we have your certificates. Ravinder Bharatidasan College. Anybody can come and collect the certificates, please. Thanks, Ruber College from Bangalore. Department of Zoology, Jamal Mohamed College. Department of Zoology, Jamal Mohamed College. Periyar Government College. EVR. Bharat College, Thanjavur. Bharat College. Bishop Nagar College, Trichy. Thank you, sirs. And I request all the participants to be seated, please. Saying thank you is not enough to capture the gratitude of my heart towards your work. I kindly request Dr. C. Nadu ma'am to thank the gathering.
Good evening to one and all gathered. As all which seems and end in life. On behalf of Department of Biotechnology, Jamal College, I take this opportunity to propose a vote of thanks to those who have directly and indirectly contributed to this conference on National Conference on Post-COVID-19 Complications, a Biotechnological Approach 2022, organized by PG and Research Department of Biotechnology, Jamal College. First of all, I would like to thank the online team for bringing the program into a grand success. And with this blessings and grace, we can able to make the entire event in impressive means. I would like to thank our vibrant secretary and correspondent sir, respected treasurer sir, and our esteemed assistant secretary sir for their motivational guidance. In their absence, thank you sir. A special thanks to our beloved principal, vice principal, and additional vice principal for their encouragement and support throughout this conference. Thank you, sir. I express my heartfelt thanks to the directors of different functionaries for their valuable support. Thank you, sir. I really extend my sincere gratitude to our chief guest, Dr. P. Tiruchirappalli, sir, Professor and Head, Department of Biotechnology, Anna University, Trichy, who spared his valuable time with us to grace this occasion. Thank you, sir. I would like to acknowledge my gratitude to all the speakers of the conference, Dr. P. Ramalingam, sir, Dr. John Bernard Johnson, sir, Dr. Kumar Somasundaram, sir, Dr. Natesan Manikam, sir, and Dr. A. Devagan, sir, for gracing the occasion and sharing their opinions for the past 2 days. We wish to express our gratitude to all our chief guests for being with us on this blended mood event. Your presence and gentle words are a source of encouragement for all of us. I express my heartfelt thanks to sponsors of this conference, Department of Biotechnology, Government of India for funding and encourage us to conduct this conference successfully. My deep sense of thanks to our judges in oral, poster, and model presentation, Dr. I. Joseph Anthony Gerard, sir, HOD, Department of Zoology, JMC, Dr. M. Salahuddin, sir, Assistant Professor, Department of Zoology, JMC, Dr. A. Ashram, sir, Assistant Professor, Department of Botany, JMC, thank you, sir, and Professor Syed Mafia Anif, sir, Anif, madam, Assistant Professor, Department of Zoology, SF, JMC. My heartfelt thanks to the coordinator, Dr. A. Sadiq Bukhari, sir, for his dedication and elegant support throughout the team. Thank you. My heartfelt thanks to all the HOD of our department and other HOD from sister department of our college. Thank you. My heartfelt thanks to member in charge, Dr. Sabashi, sir, and other faculty, Dr. S. Deborah, Abhimulika, and and valuable contribution. Thank you all. I must thank the organizing secretary, Dr. S. Ganazin. Volunteers are working hard for the last few days to make this conference very successful. I shower my heartfelt thanks to faculty and students from our college and various colleges, institutions, and universities for their active participation and so on, who spent their valuable time with us. By these gratitude to non-teaching faculty, technical support for their entire support to me. I congratulate all the winners as well as participants for their active participation. Colleges, institutions, and universities for their active participation. Once again, my gratitude to all esteemed delegates of the webinar for their presence and contribution to make this webinar a grand success. Thank you everyone once again for making it a grand success. Thank you.
Thank you, ma'am, for your heartfelt note of gratitude.
thanks to Samsudin sir, MID, Department of Zoology, for making this.
Science and technology revolutionize our lives, but patriotism and tradition frame our response. I request the gatherings to kindly stand for national anthem.
पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तब शुभ नामे जाने तब शुभ आशीष माने जाहे तब जय जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
The most beautiful day always seems to accelerate and slip beyond one's grasp just when you want to hold on to them for as long as possible. For the online participants, we will be sending the feedback form. Kindly fill and submit. The online participants, we will be sending you the feedback form. Kindly fill and submit. Thank you. The link is provided in the same meeting.
So here is a very important and a very interesting announcement. So in appreciation of the commendable performance and active participation and wonderful cooperation, tomorrow is declared a holiday for battle. Enjoy tomorrow.
Thank you, sir.
We will meet again for my next conference this week. Afra, Ifana, Suhaina signing off. Take care. Bye. Thank you all for your time.
Okay. அக்கா மீட்டிங் எட் பண்ணிருவா இல்ல அந்த ஃபீட்பேக் ஃபார்ம் சென்ட் பண்ணுமா எல்லாமே லெஃப்ட் ஆயிட்டாங்க நீ ஆன் பண்ணிட்டேன் இதுல சென்ட் பண்ண மாட்டாங்க கூகுள் ஃபார்ம்